Okay, we're gonna go ahead and get started. I just want to take a couple of minutes to welcome everybody, tell you thank you for joining us for our first virtual edition of our Rigor and Reproducibility workshop, to thank all of the speakers that are participating in this workshop. We have an exceptional team of leaders in the thought thought leaders in the space of rigor and reproducibility. You're going to hear from a couple of those this morning. We've got some dynamic speakers for the afternoon that are going to talk about data management, publication, and reporting. And our hope is that by the end of the day, you really have a greater awareness of the problem and some strategies for how to begin to address some of these issues in your own lab, in your own research, and a recognition of how different things can be depending on the type of system you work with. The intention of today's workshop is really just to be very general, to scrape the surface, to give you some resources for where to look next to find more in-depth information. I want to thank all of you that submitted your questions in advance. There were some really great thought-provoking questions that were submitted. We're going to address some of those in the experimental design panel. We certainly won't be able to get to all of them, but I do want to encourage you that if you ever have a question that you would like one of our team to answer, you're welcome to email that to me, and I can disseminate that to the appropriate person to find somebody that can actually provide some feedback on those. I want to talk just a little bit about the agenda for today. This morning, we're going to hear from Lee Ellis from MD Anderson on why this is even a problem. He does such a great job talking about how this problem really has become known, why, NIH has decided they really needed to take action. He's really been at the forefront of addressing, identifying these problems, and trying to figure out strategies for how to combat these issues. We're going to hear from Keith Baggerly after that, who was intimately involved in a case that no matter how many times I hear it, it is always compelling and just an incredible story. How this can impact, especially, the clinic, actual, people that are needing a treatment. You'll notice on your agenda, and if you don't have that in front of you, I sent it this morning, or Dawn sent it, you should have it. From 11:20 to 12, we actually have a 40-minute break. The reason for that is that we know that some of you are homeschooling kids while you're trying to work and study, and so we wanted to just give you a break to take care of what you need to take care of during that time. We ask that you're back at 12. This is where we're going to get into the nuts and bolts of the questions that were asked, and really give you an opportunity to discuss in the chat and ask questions about particulars regarding experimental design. In the afternoon, we will have the data management and resource sharing with Melissa Eitzen from UTMB, followed by publication and reporting from Stacy Gorniak. Those two sessions, we're actually going to have breakouts. So I wanted to talk just a little bit about that. I hope that when you enter your breakout session, I encourage you to have your video on. This is a great time to network, to meet new folks. You never know when you might be able to collaborate down the road. But in these breakout sessions, which should be around 10 minutes, you'll have the opportunity to tackle a case study presented by the person who is speaking during that session. Dawn is going to be sending an email. She has put you into groups. The idea is that the groups were supposed to mix up institutions, mix up career levels. You will all be in a group that's numbered 1 through 10. We have 10 groups today. You'll receive an email from Dawn, and what I need you to do when you go into your breakout room is have that email up and designate somebody to respond for your group. What we want to do is we want to collect the takeaways that your group came up with for both sessions. So we ask you to distinguish those sessions, and again, this will be explained more in the email that you'll receive here in the next few minutes. And then you will respond, reply all, and send that back to myself and Dawn. Another key thing about the breakout rooms is when you go, you need to pay attention to the number of the breakout room that you're going into. When you are prompted to enter the breakout room, it'll actually tell you you're entering room 1 or room 2. Be sure and record that because you'll need that information for a couple of reasons. One, you'll know the email you're responding to should have that information on it, but more importantly, whenever the speakers for this sessions, when we come back into the main room, they're going to randomly call on groups to report out. So you can do it any way you want. Maybe you want to have one person that serves as the one who records the takeaways and another person that's actually going to report out if your group is called on. All groups will be called on at least once. So be sure, remember, you're going to designate somebody to report out and you're going to designate somebody to reply to the email with the takeaways from the group. When you're in the breakout room, you'll have a timer in the upper right-hand corner that will tell you how much time you have left and give you a warning when you're going to be returned back to the main room. Other than that, we ask that in the main sessions, you keep your microphone muted the entire time. You can enter your questions into the chat at any time. For the morning, well, for all of the talks, actually, we'll reserve questions till the end, but you can enter them into the chat at any time. Anyway, I think that about covers it. And again, we're really glad that you've joined us. And on that note, I'm going to turn it over to Dr. Lee Ellis.
Great, thank you. Let me get my slides up here and make sure you can see them. Everybody can see the slides?
Yes.
Okay. First, I want to thank Suzanne and her team for the efforts in pulling this together. Everybody who does this makes it look easy, but I know it is not. And so thank you for that extra effort. And the effort is on behalf of the people who are here watching these lectures, listening to these talks. And I'm sorry we can't see you in person because sometimes when I talk or when Keith talks, your jaws drop or you start wiggling in your chair and you're very uncomfortable. And we're here to make you uncomfortable because this is an important issue. So I'm giving the introductory lecture. You can see my appointments at MD Anderson. I do run a research lab, but I also listed a couple extra jobs that I have. I'm vice chair of one of the NCI sponsored clinical trial groups, SWOG. So I have some input into oversight of clinical trials. I'm also on the ECOG, Eastern Cooperative Oncology Group Data Monitoring Committee, as well as 2 other data monitoring committees for trials. So again, I have some insight into what's going on in clinical trials and what's appropriate, what's not, etc. And I'm also a deputy editor of JAMA Oncology. So I do have this eye out for stuff that just can't be real or things that don't add up or things that just don't look right. So I have this diverse background. So I'm happy to answer questions at the end on anything related to these.
Lee, I'm going to just jump in and interrupt you. If your slides are not showing up as full screen, if you want to, we're kind of seeing it in the edit mode.
Okay, let me try to.
It's okay, but.
Let me work on that. And what do you see now?
Still see that. I think.
Let me stop share and share again.
Okay. Okay. And then
what are you seeing here?
Click that screen at the bottom.
My screen is at the top.
Okay.
More.
Do you see where it says notes and comments at the bottom of your screen of the PowerPoint?
Yes.
Okay, if you keep moving to where it has the keep going, past the 4 dots, the next one, click there. See if that'll do it. Yep, we're good.
Okay, sorry about that.
No,
thank you. So I'll try to make up a little bit of time. You can see how I've broken this talk down into various modules. And I'll start off a little bit lighthearted here. potential audience responses after the session, if we were using an audience response system, includes: you'll be entertained. I certainly hope so. You'll be angry. I hope you're not angry, but you should wiggle in your chair a little bit and be uncomfortable. Discouraged. I don't want you to be discouraged, but I do want you to be aware of these issues. I hope you're reinvigorated, as your mentor may be wanting you to publish in CNS. That's not the brain, but it's Cell, Nature, and Science. And you can publish elsewhere and still have an impact, and probably a little bit of all of the above. So I'm going to show you one slide. If you remember a single slide, it's going to be the next slide, everything you need to know about research integrity. And here it is. One is, I want to make you aware of retractionwatch.com. I have no financial interest in this, just to be completely transparent. I have had breakfast with Ivan Oransky, who's a professor at NYU, who runs this, but this is the email that I look forward to every morning in my inbox. And as you can see, it's up to date. Some of these are kind of funny. And then I'll come back to retractionwatch.com in just a minute. And then the ORI put out this nice little graphic for research trainees, what you need to know about research misconduct. So I'll share this with Suzanne, or maybe this can be posted somewhere, but this kind of gives you an overview of the role of the Office of Research Integrity, run by the NIH, run by the government, and how this can impact you, and you can learn a great deal by just looking over this one slide. These are just highlights that came in over the weekend. So I try to keep things up to date. This is from retractionwatch.com. You can see that it's kind of interesting is that someone retracts a paper as the authors blame the pandemic for the image issues. I think you can blame the pandemic for a lot, not sure about image issues. And you can read the rest of this, unicorn poo and other fraudulent COVID-19 treatments. Don't even know how to comment on that, but this is kind of interesting because we have way too many places to publish, whether it's in print, online, in chat rooms, whatever it is, and don't trust everything you read, of course. All right, you should memorize this. This is a list of all the COVID papers that were retracted or withdrawn as of September 28th. When I did this search, there are 33 and there are some more. And I will even highlight one. Some of these are in journals you may not have heard of, or in preprint, or submitted to preprint servers, which I'm not quite sure of their role these days, but lots of these were retracted. But one, if I can use my pointer here, was in the Lancet. And I think Keith and I were talking about this ahead of time. So the COVID era, these papers came out quickly. They may not have been statistically sound. The databases may not have been sound, but people wanted to get these out quickly. And I can tell you, as a deputy editor of JAMA Oncology, we have received a slew of papers. And the question is, number one, are they valid? Number two, is it real? Because sometimes the number of people that are reported in these cases may be 4 or 6, and that doesn't necessarily give you a firm answer. And these are some additional papers. Some are temporarily retracted. I'm not quite sure what that means, but lots of stuff going on right now in this world, and people trying to get papers out and take advantage of the disarray. Okay. Take a deep breath. Get ready to move on. So we do know that drug development is not what it should be. And if you look at cancer, which is my field of practice and research, the drug development rate, starting from concept and really moving from phase 1 trials into phase 3 trials and FDA approval, is really quite poor. It's about 5%. And we've done, you hear a lot about the drugs that have made it recently, especially in immuno-oncology and cancer. But there's lots of drugs out there in immuno-oncology which have not made it. And it's important to look at the denominator and not just the successes. We're very happy with the successes, but clearly we need to do better, and we need to have better screening of the drugs as they are being developed in preclinical studies, and then start in phase 1 where you really don't get efficacy data, and then moving to phase 2 and 3 where you do get the efficacy data and hopefully approvals based on sound data. So I'll go back to my buddy, Bob Radinsky. He and I worked together at MD Anderson for a decade. He moved to Amgen and now he's at J&J. But in 2000, after he went to Amgen, I was having some trouble in the laboratory because I had a number of postdocs try to reproduce studies that should have been led to similar results, or at least pointing in the right direction. And I just couldn't reproduce a tremendous number of preclinical studies. So I'm talking to Bob on the phone as he was at Amgen in Thousand Oaks, California. And he had told me that his boss at the time, Glenn Begley, had the same issue and had prospectively collected data from studies that were done at Amgen, where he was trying to form the foundation for the development of drugs based on sound biologic principles. And Glenn and I got together and we talked for a bit and we went over some data. And, you know, you can go back to 1994 or so. These are things called magazines. I don't know if they're out there anymore. But this is what, you know, everybody got in their mailbox once a week. And we had the cure for cancer if you were a mouse. And this has never really been fully investigated, although I've talked to several people who worked in the laboratory of the lead investigator at this time, and I have some hints, but hints should not be discussed in formal lectures. But we did a number of clinical trials utilizing the agent that was advertised in these, I call them preprint journals, if you will. And all the clinical trials completely failed. They were done in Wisconsin, they were done at Harvard, they were done at MD Anderson, and they completely failed. So how do we have tumors in mice that completely disappear, having zero effect on humans? And I can only guess at the answer to this. I don't have any proof of anything, but like I said, I have talked to people who worked up there at the time and have a little bit of insight into that. So as I was having my discussions with Glenn Begley and Bob Radinsky and others around the country, and eventually, if you look down at the bottom, the Amgen data that we eventually published in Nature, and I'll show that pop up in just a second. A number of other groups were also looking at preclinical studies, and especially in the neurologic area. And you can see here that it's pretty bad when 0 of 2 of how many studies? 221 independent studies were able to be reproduced with 70 plus drugs. So there's a major problem in cancer research, but there is an even bigger problem in neurologic research. You can say, you know, see the same thing with spinal cord injury studies. So Glenn and I got together, we worked hard. We actually had a third co-author on this who worked for a company, and his name had to be withdrawn because the company didn't want him to be part of this. But we wrote a perspective, a comment in Nature on the fact that most preclinical studies in cancer can't be reproduced, and we need to raise the standards for preclinical cancer research. And this generated quite a bit of interest, which led to meetings at the NIH with Francis Collins, Harold Varmus, I do believe, a guy that you see on TV quite a bit was in the room as well as the lead of the Neurologic Institute at the NIH. So this was quite an interesting day at the NIH director's office, and they took this seriously. And in the process, they actually changed the NIH BioSketch to reflect not all your big papers, to reflect what have you actually done. So you now have 5 subheadings of the NIH BioSketch, which ask you what have you contributed and where are the papers to support that, rather than listing just a series of Cell, Nature, and Science papers. So as stated, we were not the only ones who had trouble reproducing data. You can see Bayer, of course, a drug company, could not reproduce their data 78% of the time. And actually here we introduce a new word, irreproducibility, which, because the majority of these cases were not reproducible, but they were irreproducible. And you see a number of other authors published their percentage of irreproducible data, which is greater than 50%, which is frankly pretty sad, and most importantly sad for our patients. I do give Nature some credit. They were a pain in the butt when we were trying to submit this. They were trying to do some heavy editing, but eventually I stood firm and I said, we're not going to allow this to go through unless we send our message across. So Nature followed up on this quickly with the survey of laboratory investigators, and credit to Manya Baker, who has stayed on this, although time has passed and it's not as interesting as it was back in the first half of the last decade. But looking at 1,500 investigators that were surveyed, a majority of these thought that there was a significant reproducibility crisis, and then another 40% thought there was a slight crisis. So the vast majority of people think that there is a reproducibility crisis to various degrees. Now that's startling, because that's the first time we had ever asked people if you think there's a problem in data integrity, and most think there is. So I try to make things simple as a surgeon. I like to, put things in boxes. So here is the spectrum of reporting preclinical and clinical data. And I do believe that most people intend to be honest. And that's where the thickest part of the triangle is. But there are people who are sloppy. There are people who do selective reporting. And this has come up time and time again, only report the positive results and not report or discuss any of the negative results. And then there are just evil people, and Keith will talk about that a bit in his talk, who falsify or fabricate data. But I think over the last couple decades, since I've been in cancer research, I've seen things shift to the right. And I would like to say that this has kind of slowed down a bit since I put this slide together probably about 5 years ago. But it really hasn't. When you look at the falsification and fabrication, and I go back to that slide on the COVID publications, we ain't slowing down, as they say in Texas. So things are still moving toward the right. So what are the consequences? Well, if we have lousy preclinical data, the clinical trials are bound to fail, and we are all here for the good of our patients. And let's not ever forget that some of the consequences of our lab work, we hope, impacts patients in a positive way, but certainly not in a negative way when we're falsifying data to get our own glory with the CNS paper, but patients go on clinical trials and fail. That is not what we are here for. It's a waste of your time and your effort as investigators. It's a waste of NIH money. It's a waste of foundation money. And then we lose a loss of confidence in our community. The patients with cancer and other diseases depend upon us to put forth the best clinical trials based on the best reliable data. And if we're not doing that, we are failing our patients, which is an absolute sin. Len Friedman, who was a leader in the Global Biologic Standards Institute in Washington, became very interested in this and started putting a dollar number on this. And what he showed, along with his colleagues, was that about half of the amount of money spent on preclinical research was irreproducible. So we were wasting about $30 billion a year on irreproducible research because the foundation of that research was not valid. $30 billion a year wasted, which could go to fund you and your colleagues and people who are doing honest work. So let's talk a little bit about the spectrum, sloppy and selective reporting. And I'll put my editor's hat on. I will tell you that I always tell this to people who want to submit a paper to a journal. It gets rejected, they get upset. And I say real estate in a journal is expensive. You can't just continuously publish a journal which is 500 pages long. Nobody will read that. You have to have limits on what you publish in a journal, especially the higher impact journals, because people are expecting to read reliable information, reproducible information, and the best information. So it's hard to get papers in journals, especially the higher impact journals, because of the statement I made: real estate in a journal is expensive. So there's lots of reasons for sloppy and selective reporting. It could be inappropriate stats. And again, Keith will talk a little bit about that. Cell line contamination will be addressed later. And the bottom line is that journals don't like negative data. And I'll address this a little bit more myself as we move on. But because journals don't like negative data, principal investigators don't like negative data. I mean, they are, up for tenure, they're up for tenure renewal, they're up for promotion, they're up for awards. They want to put out good data. So selective reporting of laboratory studies, as mentioned, journals want positive results, as do PIs. So if a drug works in 2 cell lines but doesn't work at 8, are you going to publish a study which has 8 negative findings and 2 positive findings? Well, you should. But what really happens is that you only see the results from the 2 positive studies. Students, postdocs, and faculty need publications for advancement. We will never lose the quote, publish or perish. That will stay with us forever, but we have to take that with a grain of salt. And up in the Northeast, I won't mention what city or what institute, but 2 trainees will work on the same project, competing with one another. And guess who wins and gets to stay on, and guess who has to go look for another laboratory? It's the person who comes up with the exciting and positive data. So needless to say, we tend to report positive data when I'm strongly advocating that we report all the data. Recently, this came out last year in Nature, people had pushed to publish negative data. And I think that's important. And it goes back to the principle that I've talked about and will mention again and again. The pressure to publish a positive story is what people are looking for. Now, we need reviewers and publishers to allow us to publish negative results. And we do have more options now. We have open access journals. We have preprint journals. I will tell you in JAMA Oncology, if you do complete a negative clinical trial and it's well done, and there are biomarkers which help us determine, who could respond and who won't respond, I will accept that paper if it's nicely written and it's a good paper. So I'll accept negative papers. But again, real estate in a journalism is expensive. So if you want to publish a negative paper, publish it in a short version. That's my advice to you this morning. Now let's talk about misconduct. And it's hard to, I'm sure, sitting in the audience or sitting in your kitchen, it's hard for you to recognize that people actually do commit fraud. They do falsify data. They do actually, just mind-boggling to me, fabricate data. So, you have to ask the question, do people really do this, or do they just kind of massage data? So after Glenn and I published this comment in Nature, I received about 90 emails giving me some insights into what we discussed and what we brought to the public's knowledge. And these were all in support of what we wrote because of their frustration. So you look at the highlighted areas before, and in red, and in the third paragraph down, PIs wanted me to produce falsified data and I refused to do so. Many PIs fired me as soon as they realized that I don't do wrong things. To cover them up, they sabotage my professional life as well as personal character. And you'll note what I blackened out here is just too caustic for me to even show in a lecture. So people are pressured to provide positive data, and this was, like I said, one of 90, probably the most articulate of the 90 emails that I got along this topic. This goes back a few years, 2014. There was a paper in Nature that showed that by changing the microenvironment, you could convert somatic cells into stem cells. This was a huge hit until it was found to be fraudulent. They had manipulated the data intentionally in a misleading fashion. And unfortunately, after this paper was withdrawn, a senior scientist at that institute committed suicide. So this has broader relevance to the scientific community, and this is a shame, to say the least. Number one, bad science. Number two, the loss of a life because of the loss of dignity at that institute. So, does misconduct occur in the clinic? I am not going to steal any thunder from Dr. Baggerly because nobody can give as good a lecture as Dr. Baggerly, but he will wow you with his talk. And Suzanne brought this up beforehand, the story of Anil Patti. I've seen his talk probably 8 times, and I love his talk, and I look forward to seeing it one more time again this morning, because it's just hard to believe that somebody would do this. And it's hard to believe that an institute would not step in and do the right thing. So get ready. You will be seeing this in just a few minutes. Unfortunately, in clinical trials, not everything is clean either. I mentioned that with Anil Patti on the prior slide, but even here, this is not a reporter, but this is actually the Chinese government found that 80% of the country's clinical trials were fabricated, according to a report. And this is looking at 1,600 clinical trials. So I don't know what the status is currently. This is from 2016. Yeah, this was a culture where 80% of the clinical trials were fabricated. That's a problem, and that's something that brings me to another statement, which is validation, validation, validation. This is what I always tell my laboratory. When you're an editor of a journal, you look for little holes in the data or holes in reporting, etc. This was shocking to me when I saw it, but it's at this stage of my career not surprising. And what is the impact of retracted clinical papers on patients? You can imagine that, you see a positive study, and then patients receive a drug or receive a therapeutic intervention. And the Journal of Medical Ethics actually does a good job of this. So I would kind of subscribe to their e-table of contents each week. They have some really excellent papers in there. So for each retracted paper, it impacted an average of 2,600 patients per paper. And when papers were retracted for fraud, 1,500 patients were impacted. So importantly, this doesn't take into account the patients that were impacted by fraudulent or faulty preclinical studies, as I showed you early on with the Time and Newsweek journals, which had said we had found a cure for cancer, that went directly from preclinical studies without any real scrutiny to clinical trials that were done at 3 institutes and led to absolutely nothing. There are a number of fraudulent papers that have impacted patients' lives. The breast cancer and bone marrow transplants were reported from South Africa in 1999 at the ASCO meeting. This was completely fraudulent. I'll talk on the next slide about Wakefield, because I finally found an interesting reference for that. And there's an ongoing controversy at the Karolinska where Machiarina has been fired as a thoracic surgeon using stem cells to make tracheal transplants, and he's actually been accused of murder by the government over there for some of the things that he has done. But let's go to Wakefield. Now, I'm not going to read this whole thing. You can read this as I'm discussing this, but this is flu season and people are afraid to get vaccines. And what impact is that going to have on people long-term? And with COVID, people are going to be afraid to get vaccines. So if you read this story, which this was the best story, and if you look at the website, it's badgut.org. So an interesting website, but you can see how many unethical issues that Wakefield committed during this publication of a Lancet paper, which was soon retracted. He had conflicts of interest. He was developing his own vaccine that he could give to people who he could tell them that my vaccine is safe, but the other vaccines are not. He was paying kids $5 to draw their blood. I'm sorry, £5 to draw their blood. I mean, read through this. This is absolutely phenomenal. And to this date, I mean, this is 1998. To this date, people are afraid of vaccines. Now is not the time to be paying attention to articles like Wakefield's. Now is the time to pay attention to what led to the backlash of vaccines as we move into this, move through and hopefully pass this pandemic. No institute is immune. Here I'm talking about people elsewhere around the world, and you can see a number of challenges at MD Anderson Cancer Center. I list just a few. There are many more than this, but to be fair, I have to list my own institute. And Harvard, absolutely wonderful institute, many friends up there. But Harvard has its issues, and here we continue on, not disclosing conflicts of interest. That's an unethical practice. And I will add to that with a friend of mine, Jose Baselga, who was at MGH, and then he moved down to Sloan Kettering, and he failed to disclose a number of industry ties, a lead breast cancer investigator in the world. And ironically, when you don't do well in academia because of things like this, you move directly to industry, and now he's the vice president of AstraZeneca, which I find somewhat humorous, as you could tell as I'm laughing at my own slides. Hopefully you are too. One of the world leaders in cancer biology is Bob Weinberg. I mean, hardly anybody has anything bad to say about Bob Weinberg. He has trained so many big people around the country, including one of my friends, Min-Chi Hung. Senduri Mani at MD Anderson has nothing but great things to say about Bob, and Bob is has been a lecturer at ASCO plenary years ago when I happened to be in on the podium as well. Bob is probably the world's premier cell biologist, and he has had a large number of retractions, and he continues to do quite well. Doesn't seem to impact his reputation. Here are some comments after a few retractions. These are some comments online. Perhaps it's the two rogue postdocs. So cherry picking data, as I've talked about before. And, you know, they were pointing the finger at the people beneath them. And the point that I'd really like to make is that as principal investigators in laboratories, we need to keep track of our data. So here we're in the pandemic, and I see my lab's data once or twice a week, depending on how busy the week is and how my people in my laboratory are doing. But you cannot just have a Cell Nature or Science paper handed to a PI where they see the data for the first time. You need to see the data as principal investigators in real time. I want to try to stay on time here. We did a survey immediately after we put out our Nature paper. I'll give credit to Len Zwelling, who came up with this idea. And so we included postdocs in putting the survey together, and then an expert who could help us actually construct the survey. And what was interesting is that at MD Anderson, at a time when everybody's working hard, we sent out the survey to our colleagues at MD Anderson. Within 6 hours, we had 240 responses, full replies and completions of surveys, which I found was quite remarkable. And you can see the distribution here. The question is, have you ever tried to reproduce a finding from a published paper and not be able to do so? And you can look at the data and just, you know, averaging this out, being a little bit generous, you know, at least half the people and just a bit more have probably had trouble reproducing data when they've read a paper and they try to emulate that, the findings from the paper. What are the driving forces for irreproducible data? And we had greater than 90 respondents. These were trainees only. Were you ever pressured to publish findings of which you had doubt? And about a quarter or a fifth, or whatever you want to call it. Have you noted pressure from a mentor to prove that his or her hypothesis was correct? And even though the data you generated may not support the hypothesis, and 31% of people, so the mentor's, you know, idea was not necessarily, didn't come to fruition. That happens a lot, but yet they were still pressured to continue along that project and publish. And are you aware of mentors who require high impact publication before a trainee can leave the lab? And that's about 50%. So there's a lot of pressure on you as trainees to get a big time publication out there. And we need to educate not just the trainees, but also mentors on this. And then there are some selected comments. I'm certainly not going to read through this whole thing, but I thought I had a pretty good handle on the field until I read the third comment and realized that international trainees and postdocs also have visa issues. And in order for an international trainee to stay in the lab, they have to be productive or else they're shipped back home. And I know that's a harsh way of saying it, but that's the gist that I get from reading through these emails. And you know, like I said, I thought I had a handle on this, and every time I bring this up for discussion, I learned something new. So there's pressure to publish big time papers because if they want to stay in the United States and have a career here, they have to have a big time paper. And then we followed that up with Delphine and others from my laboratory. And again, I thank Keith for playing a role in this as well. And this was more recent from just a couple of years ago in clinical cancer research. And most of the respondents to this survey were postdocs, with about 10% or 11% being students. And these were all from MD Anderson. And we asked about mentor supervision. How often do you meet with your mentors? And basically, you know, most people, it was weekly, not too bad. How often do you present data outside your laboratory, perhaps at a departmental seminar series? And not too bad. You know, it'd be nice to do it more frequently to get more input, but still, you know, there's not everything I'm going to talk about today is bad. But then you get to, you know, a question of who made you feel pressured to publish positive data, and people are pressured by their PIs and by themselves to publish positive data. As I talked about on the last slide, that you have to have you know, if you're a foreign visiting scientist and you need to stay and you want to stay here and stay in the lab, you're pressuring yourself to produce positive data so that you can remain in the US. No political comments, I promise. How often do you authenticate your cell lines? I love the fact that nearly 40% of people said never. That's kind of a problem. How often do you test your cell lines for mycoplasma? About 30% said never. And I believe we'll have a discussion on this later on today. Do you and your collaborations perform blinded studies? I'm happy to see even 45% do. I know in my laboratory, when we sacrifice animals, I send back 3 people, 2 of which have no idea what group the animal was in. And God, this is going to kill Keith. Do you consult with statisticians regarding your studies? No, because I have the software. What do I need a statistician for? And that's about 50%. So there are some problems here, which could be discussed later on during this day. This is important as well. Do you feel it's necessary to publish in CNS when you're seeking your own academic faculty position? And more than half said yes. So you have to get a Cell Nature Science paper out there, no matter if it kills you or if you have to break your principles. And of course, I don't support that, but this is the reality of real life. Do you feel pressure of the publisher-pair system? Do you feel the pressure of the publisher-pair system influences the way that you report your data? In other words, you're reporting just, you know, positive data, and you're you know, burying the negative data, if you will, and it's, you know, over 60% say yes. Have you ever fabricated or falsified data? No, but I've seen a whole bunch of other people who have, and that's kind of funny. Everybody says no, but is withholding information fabricating or falsifying? It's certainly not fabricating, it's certainly not falsifying, but it's certainly dishonest if you withhold, let's say, the negative studies. And have you ever witnessed someone fabricating or falsifying data to complete a project? And again, you know, nobody ever does anything bad, but they certainly do see it in other people. I want to bring to your attention Elisabeth Bick. For those people who are on Twitter, and I'm on Twitter, this is a postdoc who worked with 2 microbiologists, Veric Feng and Arturo Casadeval, who are members of the National Academy of Sciences, and I've had the pleasure of working with them on research integrity. And basically, she has a photographic memory. So she can not just look at, figure 1 and figure 7 and see that they use the same, control bands for Western blotting, but she can remember a paper she read 3 years ago and the figure in that paper and note that there's something, that I've seen before. And she has this incredible memory, and she can go back and find that people have used the same images or manipulated images in ways that most of us would not be able to recognize. And she will put out tests on Twitter to see if you can find the fraud, if you will. Just to look at this, she did an assessment of the percent of papers with potential image duplication or image manipulation. And this can be as high as 11% in some of the years that she has gone back and looked at the papers, and especially in the field of microbiology. So why does this occur? I've talked about this a lot. You have a strong mentor. If you're a trainee, you have a strong mentor and they're pressuring you. Sometimes it's because you need to stay in the country and English is a second language. It's hard to rebut your mentor if you are, if English is your second language. You need high impact publications. I've stressed that and shown you data on that through all along. And you can't leave the lab until you have a high impact publication. Well, faculty have stressors as well. We have to publish or perish. We have to get our tenure renewal. We have to get promoted. Endowed chairs help tremendously in funding a laboratory. And to do that, you have to have those CNS papers or do something similar, either in service or in administrative or some other way. For grants, you need really strong preliminary data. All of us suffer the 9% funding rate right now at the NIH. And also, it's great to walk around the halls and everybody congratulate you for your recent Nature paper. And as I've talked about before, you can't ever leave out financial issues. There are patents and sub-licensing if you identify a drug or a new biomarker or a new approach to treatment. You can personally gain from this. It goes back to the Nature survey, which was very similar to what I've just discussed. I'll focus on the right side of the slide here. What factors contribute to irreproducible research? And I've gone through this before: selective reporting, the pressure to publish, poor statistics or no statistics, etc. So you can read this yourself. Even though it's an old survey, I still think it's a relevant survey to today. So let's talk about high impact publications and what this does to our culture. And the quote a postdoc at the South Campus of MD Anderson, who was walking through the hallway. I ran into her when I was an interim chair of cancer biology, and she turned to me and was a little shaken and said to me, "My mentor just told me you are nothing unless you publish in CNS." And that's a bit of a shocker. She was not happy with this this pressure, but I talk about this pressure. I've probably mentioned it 30 times already in this talk. Going back to my friends Arturo and Ferric, I'll just highlight the point of this abstract here is that the impact factor mania, a term which you probably will remember, is associating the value of the research with the journal where the work was published rather than the content of the work itself. The mania is causing profound distortions in the way science is done that are deleterious to the overall scientific enterprise. And I completely agree. We have a hallway at MD Anderson where if you have a big time publication, you get a picture taken of yourself and your lab and the publications put up on the wall. But then there could be a +1 paper, which really has a bigger impact, which we'll never get on the wall because the impact factor of +1 is nowhere near the impact factor of Cell Nature or Science. And then there's a question, should we eliminate the impact factor? What's interesting is that, again, Fang and Casadevall did this analysis, and they looked at a retraction index, which they created, and they found that the higher the impact factor of the journal, the higher the retraction index. And this can also be attributed to the fact that, yeah, when you read New England Journal of Medicine, there's a lot more people who are reading that. It comes under more scrutiny in some of the less often read papers and less often read journals. but it still sends the message that just because it's published in Cell, Nature, Science, or New England Journal of Medicine doesn't necessarily mean that everything in there is true. And I've talked about the Wakefield vaccine fiasco, etc. So I do like to throw in a little philosophy here attributed to Confucius. A man who has committed a mistake and doesn't correct it is committing another mistake. So hopefully people learn not to do this. And if they do it, let's hope it's only once, either because their career is over or because they've learned their lesson. I want to skip through this in the interest of time, but you get the point here. There are some people who really do believe in avoiding impact factor mania. And my final comment is there's nothing wrong with publishing in Nature, Cell, Science, or whatever your preferred high impact journal is, but never compromise your ethics to do so. So how do we fix this? Okay, memorize this. This was a review article I wrote back in 2015. I was awarded the John Wayne Clinical Cancer Research Lecture at the Society of Surgical Oncology. And I don't think much has changed here. I think that, I highlight, allow the submission of negative data, allow some feedback. We can talk about pub peer some other time. We don't have time to do that today, but I think that we do have to have a culture change, and I think I've beaten that into your heads for the last 40 minutes or so. Let's talk about the Office of Research Integrity. I introduced this earlier today, and what's disappointing to me is that these are the number of case reports where people have been absolutely found to be guilty of committing fraud in scientific research. And what I'm disappointed in is that there's not more, because I know there's more out there. But these are very thorough investigations, and these people have some penalties, which I'll talk about on the next slides. But that's very few for the entire United States, with all our grant funding mechanisms through the NIH, the DoD, or whatever. And each institute that you work at has a research integrity office, and you can contact them directly and even anonymously. But what are the actions? What if you have a paper, what if you are found guilty of fraud? Well, the paper has to be retracted. That's a no-brainer. You have to have your research supervised for 3 years, which I have no idea what that means. You can't serve on committees for 2 to 3 years. So you get rewarded for not, for being dishonest. And most can still receive NIH funding. And this is mind-boggling to me. So I think the punishment has to fit the crime. I'll let you read this on your own, but I think we need to revamp the system and make it a little tougher. I mean, one shot and you're out. And that would help kind of limit the number of papers that come out which aren't reproducible. The primary inquiry rests with the NIH-funded institution. So if you go to the ORI website and you think that somebody is committing fraud and you're brave enough to, submit that name, whatever it is, the ORI will come back to your institute and say, you need to investigate this. So the primary responsibility is with the individual institutions, and that creates a conflict of interest. Although I would say that the people who are conducting these investigations are as honest as can be, and Bill Plunkett is a friend and one of the most honest people who walks this earth, so I have no problems there. But perhaps there are institutes where people are not as grounded as someone like Bill Plunkett. So the allegations can be brought to anybody, and there's an information gathering meeting and initial fact finding, and but these are your own peers evaluating whether or not you committed fraud. And if you do think that fraud is ongoing, but there's no one at your institute, most universities or university systems, like the University of Texas system, have a website for abuse, fraud, and/or unethical behavior. So if you're a little fearful of having your emails tracked, whatever it is, you can go to a anonymous website. So I'll end here. I'm a Beatles freak, and in my background on Zoom, you'll see a picture of John Lennon recording Imagine. This is above is a quote from a friend of mine, Mike Fish, after I published that article and paper that I talked about before. But I love John Lennon's quote, which is, "Give me some truth. I'm sick and tired of hearing lines by seasick, narrow-minded, short-sighted hypercritics. All I want is the truth, just give me the truth. And that's the way we should view research. And Albert Einstein was no slouch himself. He stated here on the outside of the National Academy of Sciences, the right to search for truth implies also a duty. One must not conceal any part of what one has recognized to be true. So these are two of my heroes. I hope I've selected good heroes. I think I have, and I hope you live by these principles. And
Thank you,
John just had a
Lee.
birthday.
Appreciate that. We've got a couple of minutes. I'll invite you to feel free to post some questions in the chat. Wait a couple of minutes for that. While we're doing that, I have just one thought question, and that is, you know, we've got a few years under our belt of knowing this is a problem, and journals and NIH, you know, having requirements around publications and grants. Any studies to actually determine if there's been any improvement in this area?
I have not seen any. I mean, I do these weekly searches that come in my inbox Wednesday mornings on research integrity and colon cancer biology, et cetera, and I really haven't seen anything. Also, of course, retractionwatch.com would probably be the best source for that. You know, it looks like it's a flimsy or a funny website, whatever it is, but that's why I mentioned that Ivan Oransky is a professor at NYU, and this is a completely reliable, you know, source of information, and in my opinion, and they will go back and give you the backstory. But I have not seen any metrics on this. You know, we kind of hit a peak about 2014 to 2016, and everybody was interested in this, and then it's kind of gone away. And of course that occurred, as Keith will talk about, you know, during a time when there were lots of ethical breaches, et cetera. So really not much has been done. I have not, and again, the number of people who have been found guilty of fraud by the ORI has gone down, which frankly is surprising to me, considering the funding rate at the NIH right now, et cetera.
Yeah. We've got a question here from Curtis. Is it such a great thing to publish negative data? Isn't negative data maybe more prone to procedural error?
So good question. I'm assuming that it's good to publish negative data if everything was done right. In other words, being a colon cancer investigator, if you look at 10 cell lines and 3 cell lines, a drug worked, and 7 cell lines, they didn't. There may be something in the 7 cell lines where a drug didn't work, such as a BRAF mutation or a KRAS mutation. So you should be looking at the negative data and saying, okay, why was this negative? It's not necessarily because of the experimental conditions, but because maybe there's something inherent, you know, to those other 7 specimens, patients, cell lines, whatever it may be. So publishing negative preclinical data, I agree, that's a challenge. That's an uphill battle, but I think it is important because in the negative materials, there may be something which is a biomarker. And again, I'm talking as a cancer investigator. This may not hold true for other areas of interest that, you know, are of interest to the people and their specialties on this, in this class today.
I see. So you're saying that it needs to have a positive control for sure in order to validate.
Well, I mean, if you know, do a study and yeah, you need to have a positive control. If nothing works in 10 cell lines, then, you know, is it because you didn't do a good job, or is it because there's something about the 10 cell lines and they're all BRAF mutated and therefore they're all resistant to XYZ therapy, whatever it is. So yeah, you probably do need to have a positive control to make sure that you know how to do laboratory research. Good point.
So along the lines of, you know, kind of with the negative data, we have a question from Michaela. She notes that, you know, is it possible the relationship between impact factor and retraction index is due to higher surveillance of higher impact factor journals? She also notes that I've heard publishing negative data is important to ensure rigor and reproducibility for some time now. However, I haven't heard any initiative to incentivize it. Is there anything actually being done to help the problem, or just reporting on the issue?
So for the first question, I mentioned that in Arturo and Barrett's article. where the greatest number of retractions occur in New England Journal of Medicine, et cetera, which actually, frankly, I think that data is old. I don't agree. And it is because higher scrutiny. I mean, everybody's reading New England Journal or Nature or Medicine. For the second, I'm sorry, repeat this, Suzanne, repeat the second part of the question.
The second part is, even though it's, often stated that publishing negative data is important to ensure rigor and reproducibility, they haven't heard any initiative to increase or incentivize it. So is there anything actually being done to help with that?
Nothing is actively being done to incentivize that. What I would recommend is that when you publish positive data and you have negative data, you include that. So I think you will get some points for being thorough. And again, that opens up an opportunity to figure out why some of this stuff didn't work. But I think it is our own moral responsibility to publish the negative data. I can tell you from a clinical trial point of view, I myself will publish negative data, but I don't want to take up too much space in a journal. So I will cut that down to 1,200 words with a whole bunch of stuff in a supplement. For preclinical data, yes, as discussed, you need to have some positive data, but you need to throw in that negative data and not just throw it away, but throw it in a supplement, put it in the paper, and note, this drug did not work in all cell lines or did not work in all models. And I think it's upon us to do that. I don't think you will get dinged, if you will, by a reviewer if you state that not everything worked, because we all know not everything works. If everything worked, we wouldn't be having this workshop. We'd be out there celebrating the cure for cancer or cardiac disease. So I think it's upon us to state that it worked in 2 out of 5, cell lines, or hopefully, 4 out of 10. You get some statistical significance there for Keith. But it ultimately falls on our shoulders or the mentor's shoulders to teach that to the lab. Thank
you, Lee. I'm going to note that we have several really good questions in the chat, and invite Lee, if you have time, you can click on the bottom of your screen on chat and just answer those directly in the chat. But for the sake of time, we're going to go ahead and move on to Dr. Baggerly, who is going to talk to us about this great story you've heard us preparing you for. Keith?
Hi there. So give me a moment to get everything shared with everybody, and we will begin. So let's see if I can do this right. And oh dear. Okay. Okay, can everybody see my slides?
Yes, we can.
Yes, we can. Okay, wonderful, wonderful. So as Lee has been telling you, he's been describing one aspect of the reproducibility story. And I am going to come at it asking the same question, why are we talking about this? But I'm going to be coming at it from a slightly different angle. Specifically, I'm going to be coming at it from the point of view of a numbers geek or a data scientist. See, I am a statistician by training. I worked at MD Anderson for about 20 years, and I had the honor or good luck to start at MD Anderson right at what I will describe as the beginning of the data tsunami of high assay data. What I mean by this is the emergence of things like proteomic assays, microarray assays, various other things, where given a sample of interest, we can measure it several thousand different ways, get several thousand different numbers, and that's a ton of information. And that got people really excited because there is this underlying hypothesis that if we've got this much information, then the clinical questions should be answerable from this somehow. We just have to figure out how to look at the data the right way. So we've been trying for the past several years to look at data the right way. And that's when reproducibility gets to be important. But there's a further aspect to this, which is that in some ways, the questions actually become harder when you deal with hundreds or thousands of measurements than just 1 or 2. And that's because, well, quite frankly, I know several clinicians where if I go to them and I say, you know what, when the expression level of this gene goes up, the patients do poorly, and they'll say, yeah, we know that, and they'll give us the clinical backstory. They can come up with a clear explanation. And they'll be right because they've got good intuition. Nobody has good intuition when we start talking about signatures of 50 or 100 genes or more. We have presented lists of this many genes to clinicians, and it's wonderful. We will see them inventing a story right in front of our very eyes, where they'll say, yeah, that must make sense because this interacts with this and this interacts with this. And you will see a story emerge, and it is beautiful, but I don't believe it. I don't believe it because I've seen it be flat wrong. So that presents us with a conundrum. We really want there to be the possibility of finding a story and clinically using all this information, but we can't trust our intuition to be a good guide. So what that forces us into the position of is saying, we've got to be able to track every step by which this was assembled, where the steps are things that we can check. That's the level of rigor that we have to impose just to be sure that we're doing it right. Now, if you take this idea and you then go to the literature and you read papers and you say, oh, this is wonderful. I think this is a cool idea. I know before I invest too much time of my lab's resources, I'm going to sit down and I'm going to try to replicate their findings using the data they came up with. That should be easy, right? Because they describe the methods, the data in their paper, and it's already outlined there. We've tried this a few times. And unfortunately, I can tell you that it is not always easy to do. And you will discover some steps that are missing. You will discover some cases where it's hard to find the data, etc. And well, in some cases, when the paper is really important and you can't quite figure it out, this forces you to do something different. So in particular, we were led to invent a brand new subdiscipline of bioinformatics, which I call forensic bioinformatics. And what this is an art. This is an art of saying, here's the raw data they started with, here's the results they reported, what the hell must they have done to get from here to here? And that type of reconstruction is often informative, though not always in the ways one might expect. And I'm going to take you through a story, an illustration or a case study in this case, dealing with a clinical question that's of pretty large interest to folks at MD Anderson, which is, can we predict which patients will respond to which chemotherapeutics? Why is this important? I'll tell you it's important because if a woman shows up at MD Anderson today with advanced ovarian cancer, we will give her standard frontline therapy, which is a platinum agent and a taxane. We will give her this therapy knowing that for 80% of the women we give this to, it's going to help them. The tumors will shrink, they will feel better, their quality of life will be improved. That's the 80%. The other 20% we will poison for 6 weeks to no good effect. We will do it because we don't know what 20% they are going in. If we did, we'd try something different. And it was against this backdrop that a paper appeared in Nature Medicine several years ago that got a lot of folks really excited because the authors claimed to have largely solved this problem. They said, here's what we figured out how to do. We figured out how to use information from a brand new high throughput assay, and at this time it was microarray data, to make such predictions. We're going to do this by starting with data on a bunch of cell lines. And in particular, we're going to start with a panel of cell lines that's been maintained by the National Cancer Institute for the past 30 or 40 years. And this panel is known as the NCI 60. Now, what's special about this panel? What's special about this panel, and the reason the NCI has kept it around for so long, is that one of the ways that we understand how various drugs work is every time we get a new chemotherapeutic in for exploration, the NCI will take that drug and they will assay it for effectiveness against every cell line in this panel. Specifically, what is the dosage of this drug that's required to cut the growth rate of this cell line in half? And thus they can assemble a profile of sensitivity of each of these cell lines to each drug. This information of sensitivity to each drug for all of the cell lines in the panel, this is publicly available data. So if you can figure out some way to extrapolate from how cell lines respond to figure out how patients might respond, then this data set could, in theory, serve as a Rosetta Stone to make predictions for any patient we've got based on any drug we've got. So, okay, really cool. So how does it work? Well, they started off by going to this panel of cell lines, and they looked at the microarray data for these cell lines. Again, this is publicly available data. And they said, let's take a drug of interest. Let's say it's docetaxel. We will use the sensitivity information to sort all the cell lines in this panel from most sensitive to most resistant. And then we will take the profiles of just the most extreme ones, the most sensitive and most resistant. Let's say 7 cell lines at each end. And then we will compare the microarray profiles of those extreme ones, gene by gene, saying compare sensitive to resistant using immensely sophisticated statistics like 2-sample t-tests to say which differences are the biggest. And then, having identified which ones are the biggest, we'll take the 50 genes that are the most different, and we'll say, let's focus on those 50 genes because the pattern of response in those 50 genes, that is the pattern of sensitivity to docetaxel. If we then look at a patient's profile and we say, does the profile for the patient look more like a sensitive profile than a resistant profile, we'll say, give them the drug, it'll probably work. If it looks more like a resistant profile, we'll say, let's try something else. So that's how it works in theory. And the reason it got into Nature Medicine, got a lot of people excited, was they said, we've tried this approach using publicly available data sets for 7 widely used agents, and we got great results every single time. So what does this mean? This meant that within weeks of this paper appearing, we had lots of different PIs at MD Anderson coming to us in bioinformatics, pointing to this and saying, we want to do this here. We want to treat our patients better. We want to use this method. But this method involves math, and that's scary. So we need your help. And we said, okay, that's fine. That's wonderful. Because we are helpful bioinformaticians, and this looks really cool to us. And furthermore, because all of the data they're using is publicly available, we should be able to check it ourselves and be able to build on it. So we said, all right, let's try this. And we're going to try working through what they did in order to make sure that we can use it. And to test this, the very first question we set ourselves was something we thought should be easy, which is for one of the drugs that they looked at, let's see if we can identify the genes that were used and see if the list we come up with matches theirs. We thought this should be pretty easy because as a supplement to this paper, there's a list of tables saying, for this drug, here's the genes we found. For this drug, here's the genes we found, along with some explanation in the paper about why some of these genes make sense because of how they operate in terms of biology. So we said, okay, great, that's wonderful. We know this should be easy to do and we can check it. So we said, all right, let's start with a drug, say 5-fluorouracil. It's one of the ones they picked. We identified the most sensitive and the most resistant cell lines. I did the 2-sample t-test myself. I am a PhD level statistician. I trust my ability to do this. They reported 45 genes. I got my top 45 and we checked for the overlap, and the overlap was 0. Nada, nothing, nil. And we said, that's probably bad. So what's going on? Well, one thing that's going on has to do with the fact that when you start dealing with not 1 or 2 genes, but hundreds or thousands, you often don't refer to them by name anymore. You start giving them abstract indices simply so that you can keep track of them. And in this particular case, the arrays that they were using were made by Affymetrix. They're known as Affymetrix gene arrays. And this comes up because the things that identify genes on these arrays are probe set identifiers. So here are the first few probe set identifiers that they identify as being important in response to 5-fluorouracil. Here are the first probe sets that we come up with. If you go from their list to ours, you notice a pattern here? We have an off by 1 indexing error. How did this come about? Well, it turns out that the software that they're using requires 2 inputs in order to work. The first input is a matrix of gene expression values, which is genes in the rows, samples in the columns, with a header row saying, is the sample sensitive or resistant to treatment? The second input is a list of gene names in the same order as the expression matrix, but for their software to work correctly, this list of gene names must not have a header row. So if you're working in Excel and you cut and you paste and you leave on that header row, guess what happens? You get an off by 1 indexing error. And guess what this means? It means that you will come up, or you'll look at a signature for 5-fluorouracil that appears to work, and you'll come up with an explanation in terms of the biology of genes that aren't involved at all. And they came up with a story. And they came up with a story for every one of the 7 drugs they looked at, because this mistake is there for every one of the 7 drugs they looked at. And it got by the authors, it got by the reviewers, it got by the editors, because our intuition in hundreds of dimensions sucks. So this is a problem. But as we stared at this and we said, okay, not happy, something occurred to us, which is that this problem, while it's embarrassing, is actually not fatal for what they want to do. What they want to do is they want to predict response. And that's actually a characteristic of the underlying numbers, not the gene names. The gene names are things they tacked on at the end to make sure they understood what they were dealing with. But if the numbers still allow us to separate responders from non-responders, that's still something we can clinically use. So let's take a look and see, can we take the numbers they're working with and can we find separation? And we said, all right, let's test that ourselves. And again, we want to set ourselves a bar to say, are we doing it right? And we decided to say, we understand things well enough to use it ourselves if we can get predictions at least as good as the ones that they got, because those were good enough to get them into Nature Medicine. So those must be pretty good. So that leads to the question of how good are their predictions? So let's take a look. This panel in the top here, this is a panel showing the scores that they came up with for 24 patients, breast cancer patients, who were treated with single agent docetaxel. Most of the patients who responded to treatment, who are sensitive, they're blue squares and they have low scores. That's good. Most of the patients resistant, red triangles, they have high scores. That's good. There's clear separation between sensitive and resistance. But here's another thing that comes up with data analysis. And I wish I could see the audience, because one of the things I like to do is I like to ask questions. So I'm going to ask you a question, but I'm going to still show you the answer here because I can't really watch the audience, which is, how many squares are there in this plot? So we actually went in and we counted. 1, 2, 3, 4, 5, 6, and we counted and we got to 13 blue squares and 11 red triangles. We have 13 sensitive and 11 resistant patients. Now, why does this matter? It matters because I told you that all the data that they're working with is publicly available. So we actually went back to the source paper for this data set, and this was in the Lancet a few years before. And in this paper, the authors said, well, if you want to tell our sensitive from our resistant patients, you should focus on contrasting our 11 sensitive with our 13 resistant patients. 11 and 13, not 13 and 11. And that had us rather disturbed. So we said, okay, we don't necessarily trust this drug. What about another one? Well, here's adriamycin. And for those of you in the audience who are real overachievers, how many red triangles are there? For those of you who don't want to go there, you'll probably give me that it's mostly red, so that we've got a whole bunch more resistant than sensitive. But again, they claim great predictive accuracy. The problem is that this is a paper taken from the New England Journal dealing with the treatment of childhood ALL, a form of leukemia. And the reason this is relevant is that as cancers go, this is one we know how to treat. We've got about an 80% cure rate here. And what this means is that if in this patient population, we have a drug to which most of the patients are resistant, we wouldn't be using that drug. So what's going on? Well, the authors of the New England Journal paper thought that most of their patients were sensitive, not resistant. So we went back and we looked at the software again. I told you before that it required 2 inputs, and that one was a matrix of gene expression values with a header row, and the header row should say sensitive or resistant, but that's not quite what they did. See, we have the gene expression matrix, and the header row, for the header row, they used 0 and 1 to indicate the 2 groups, but they never explicitly wrote down 0 equals sensitive, 1 equals resistant. So in some cases, as they went from one drug to the next, they got the 2 meanings reversed. So what does this mean clinically? It means that if we take their recommendations and we were to apply it, we would in this case be administering adriamycin only to the patients who are most resistant to it. This is poor clinical practice. This is a bad idea. So we are not too happy about this. Now, all of this back and forth and looking at the data and things like this actually is not the result of just a 10 or 20 minute skim of the data. This is actually the result of about 6 months of us going back and forth with the authors of the paper saying, we think your method is great, we want to reproduce it here, we'd like to check it. By the way, are you aware that the gene names are wrong? By the way, are you aware that the sensitivity labels seem to be off? And then we had to tell them that, by the way, when we use the labels which we think are correct, we don't see the separation you report. Can you help us? And at this point they said, well, obviously this collaboration has ceased to be productive, so we wish to draw it to a close. And we said, well, okay. So we wrote up our various objections and we submitted it to Nature Medicine. And this appeared about a year after the initial article as a letter in Nature Medicine. It appeared together with something we hadn't seen, which was the author's rebuttal. And we were really curious what they were going to say, because we said, okay, here's our outlining of the errors. Here's 100+ pages of documentation saying how we found the errors. And were they going to say, oh my gosh, we're so sorry, we'll never do it again? That wasn't quite the tone of the report. The reply was that, unfortunately, Doctors Baggerly and Coombs, their analyses are deeply flawed. We have gotten great results. Our labels are correct. We've posted data to our website to show this. This is including the things for the sensitivity labels. And furthermore, since our initial Nature Medicine publication, we have continued to apply this approach in dealing with other high-profile studies, getting more publications and more proof of principle. So we must be right and they must be wrong. Well, I found out about this the night before I was supposed to give a talk on this at the NCI. So the night before I'm supposed to give a high-profile talk, I'm finding out that everything I'm supposed to talk about is wrong. This did not put me in a good mood. But okay, they say the data shows this. Maybe we screwed up. Let's look at the data. So went to their website, downloaded the data they posted there for adriamycin. That's the one with all the red triangles. Looked at the data. It's a big Excel table, big matrix of data, 22,000 odd genes, 144 columns, which are samples. The first 22 columns are for cell lines, and they're labeled as such. The next 122 are patient samples. They're labeled as such. And in addition to saying, is it a cell line or is it a patient sample, they say, is it a responder or is it a non-responder? So for the patients, we count the number of non-responders, red triangles, and we get the number they published. We said, well, darn, maybe we screwed up. So maybe there's some pattern in the data. So we looked at the data and we said, if we look at the columns of numbers, can we find patterns? Can we find some that are closer to others so that we can try something that might tell us about what's going on? And when we started looking at patterns and how close the samples were, we saw something that was pretty weird. So what I'm showing you here is a plot of the pairwise correlations of all of the samples here, where the correlations are really high, essentially 1. Down here, we're contrasting cell lines with each other. This line of red dots on the main diagonal, that says that cell lines are highly correlated with themselves. I believe that, that's good, okay? Out here, we have the patient samples. This line on the main diagonal, that we expect, cell line, or patients are correlated with themselves. These other red dots, we didn't expect. What's going on? Well, as it turns out, they have 122 columns of patient samples, but if you look at the numbers, not all 122 columns of numbers are distinct. Some columns of numbers have been reused 2, 3, or even 4 times, so that they don't have 122 distinct patient samples there. Furthermore, right about here is one patient whose data was used 4 times, where that one patient was labeled resistant 3 times out of 4. So we're pretty sure they don't have the labels right yet. And we sent a note about this off to Nature Medicine, and Nature Medicine said, well, the authors have gotten back to us, they'll make a correction, there's no need to publish your objection. So we said, okay, we'll wait and see what they publish. And indeed, another year later, they published their reanalysis using only the 95 unique samples, and they got great results again. The problem is that when we counted, we'd only seen 84 samples, not 95, so that was a surprise. And when we looked at the 95 they said they were using now, only 80 were distinct, 15 were present twice, and of the 15 present twice, 6 were labeled both sensitive and resistant. So we still thought that there was a problem. The correction of a correction still needed correcting. But Nature Medicine had said it was done, so we moved on. We moved on in this case to look at one of the papers they said that they'd used to validate this approach. And this was a paper that showed up in the Journal of Clinical Oncology. And in this case, they'd extended their approach to deal with some new drugs, cisplatin and pemetrexed. And cisplatin is used really widely. And they said, we've found signatures for these drugs. And furthermore, these make biological sense because using the new arrays that came out this year, these have more genes from the genome on it, we find in the signature for cisplatin ERCC1, ERCC4, and DNA repair genes, and these look biologically plausible to us because there had just been a series of papers in the New England Journal talking about this is how cisplatin works. So people were really excited, and the lung cancer folks asked us to say, could you check these signatures? Can you reproduce these? And with some work, we were able to match all 80 genes that they reported for pemetrexed. After we accounted for the off by 1 indexing error they were still making. For cisplatin, they reported 45 genes. We looked at them. We were able to match 41 of the 45 genes after accounting for the off by 1 indexing error. For those of you good at subtraction, of course, that leaves 4. There are 4 genes they report as being important for cisplatin that the data don't give us, that we can't match at all. So what are the 4 that we can't match? Well, it turns out the probe sets that we can't match are for ERCC1, ERCC4, a second probe set for ERCC1, and another one for FANK-M, which is DNA repair. These are the genes whose presence in the cisplatin list they use to cite for biological plausibility, but the data don't give those. So we looked at that and said, okay, that's a problem. But even more, even in this context, these last 2 probe sets are special. Why are they special? Those probe sets are special because they weren't physically present on the arrays that were used. These arrays were part of a 2-chip set to cover the entire genome, but since the U133A had more of the known genes, people tended to run this one and not the 133B. So as it turns out, 2 of their best genes they've never measured yet. We thought this was a neat trick. So we wrote up a note about this and sent it to JCO, and JCO wrote back and said, we're sorry, we can't publish this. We sent them a second note saying, could you tell us why? And they sent us a second note saying, we're sorry, we can't publish this. So we said, okay, let's move on. We moved on next to the other paper cited as validation, and you can see it's right there in the title, validation of gene signatures. And indeed, what they did in this case was they looked at data from a large clinical trial run in Europe where they expanded the approach to deal not just with one drug, but with combination chemotherapies. And this is important because in cancer, we don't typically treat cancers with just one drug. We try to hit the tumor several different ways. So what they see? Well, in this particular case, these breast cancer patients were allocated to one of 2 treatment arms. The 2 treatment combinations were either fluorouracil, epirubicin, and cyclophosphamide, that's known as FEC, it's a 3-drug combo, or Taxotere, followed by epirubicin and Taxotere. This is a 2-drug combo. The key point, the key finding of theirs is that they say that if we'd used their approach to allocate patients to treatment arms rather than randomizing, which is what was actually done, we would have improved the response rate to these therapies in this cohort from 44% to 70%. That's a big deal. So we wanted to know, can we get this to work? Can we try this here? The breast cancer folks at MD Anderson asked us about this. So we said, okay, let's get the data. So we downloaded the raw array data and we said, do we see 2 clusters? Say the ones that correspond to TET and FEC sensitivity. So here, I'm showing you a plot, again, of correlations. Now they're not quite so extreme, of all the array data we downloaded for this study. And the fun thing is that we didn't see 2 clusters, we saw 3. And we said, what's going on with these 3 clusters? And we looked and we said, well, actually, if you look at the array data that's posted, these come in cell files, that's the abstract name for them. And one of the things that's really cool about these files is that in addition to saying, for this gene, here's the expression level, they also have some metadata and this metadata includes something like, what day was this assay run? So over on the right, I've plotted these arrays as a function of what day they were run. This first block is right here. The black circles, those are patients who were treated with FEC. Green X's, those samples were excluded from the final analysis for one reason or another. This second block, that's this block in dates. That's the second half of patients treated with FEC. This third block, that's all up here, red stars, that's all of the patients treated with TET. So what are we seeing here? All of the patients who were treated with one drug combo, specifically TET, had their assays run several months before all of the assays for all the patients treated with FEC. And as it happens, it's not just several months before, these assays were run in a different lab in a different country. So what's going on here is that what we have is complete confounding of the experimental design. in addition to changing the treatment, we've changed several other factors going from here to here. So we will pick up some differences, and some of those differences won't be due to the biology of interest. This type of complete confounding, this is a common screw up in very many studies, and this will be talked about later in the course today. But here, what this meant was that, well, we weren't too happy with this study, and we weren't too sure whether we could use it. So we said, all right, is there something else we could do? Well, they're using this to make predictions for combination therapies now. And we were curious as to how they did that, because we'd sort of understood how they were making predictions one gene at a time. So how did they put them together? And this is where we started to put our math geek hats on. And I'm going to apologize because this next slide is going to be the only one of this entire talk with formulas on it. So what we said was, to see if we can understand how they're doing it, we went back to their initial Nature Medicine paper, and they outlined this idea there. They said, well, we think that this is how it might work, and they outlined how they might combine drugs for a 4-drug combo, TFAC. So we've actually got combinations for a 4-drug combo, a 2-drug combo, and a 3-drug combo. So what are the mathematical rules that they are using? Well, we figured them out. Here's the first rule. The probability in the 4-drug combo case, the probability that a patient is responsive to treatment with this 4-drug combo, they say is given by the probability they're responsive to this drug plus the probability they respond to this plus P of A plus P of C minus the product of all 4. And we stared at that and said, that can't be right. And indeed, the authors said that can't be right because all of the probabilities they got using this rule were bigger than 1. And they said, well, okay, we know that that's wrong. We know the biggest one should be 1 and the smallest one should be 0. So we'll set those and linearly interpolate everything down from there. This interpolation step didn't make it into the methods section, but we figured out that's what they did. So we looked at that and said, okay, we don't like their math. But still, it's what they're using. Now, what about the other combinations? Well, actually, for a 2-drug combo, this rule is okay. It's the independence rule for the combination of events. It's just they generalized it to more than 2 improperly. So what is the rule that they actually used for the 2-drug combo? This does give them numbers between 0 and 1, but the rule is different. Well, what about the 3-drug combo? Here's the rule they used. I have a standing offer to buy dinner for the first person who can tell me where on earth this rule comes from because I have no clue. I can simply tell you it's what they used. But what I can tell you is that each rule is different. Now, as a bioinformatician, to paraphrase a rather famous American, I have a dream. And that dream is that if you're going to validate a biomarker signature going from one study to the next, that you would keep that signature the same. But that was not done. So we said, all right, that's rather frustrating. Is there anything we can do with this paper? Well, we don't like the design. We don't like the combination rules. Is there something else? Well, maybe if we just looked at the samples 1 drug at a time, we could find something useful. And indeed, the authors thought that that was probably the case. And they said, if you look at just the results that we get for Cytoxan, one of the drugs, and you plot our ROC curve to see, can you tell defect responders from non-responders, we get an almost perfect ROC curve. Using the results from just this one drug, we can do a great job. And we looked at this and said, huh. So we downloaded the data and constructed an ROC curve. And this is the curve that we came up with, which looks a bit different. And then we stared at that and we said, well, wait a minute, there's a different question that comes up. And this question is not mathematical, it's biological. The biological question has to do with the nature of Cytoxan. You see, Cytoxan is what's known as a prodrug. It's not something that's active in and of itself. You give it to a patient and their liver processes it into its active form. So what do you think happens if you give a prodrug to a bunch of cell lines that don't have livers? Nothing whatsoever. How did they identify cell lines as sensitive and resistant to Cytoxan in the first place? Well, we tracked down the cell lines that they used, and it turns out that they are sensitive and resistant, but it's to pemetrexed. They got the drug wrong, but they got great results, so who cares? So we wrote up all of these findings, and we sent these off to the editor of Lancet Oncology. The Lancet Oncology wrote back that unfortunately you are arguing about statistical issues to which, as everyone knows, there are no right or wrong answers. And as a result, we don't need to publish your objection. And we breathed a deep breath and moved on. We moved on next to look at glioblastoma, or brain tumors. because they published another paper where they said, using temozolomide, we can come up with great predictions here. And we looked at, and our brain cancer folks came to us and said, can you look at this? Can you check this for us? And by this point, we thought we understood it. So I said, sure, we'll try. We downloaded their data. We tried to reconstruct their signature, reconstruct this heat map, and we tried for weeks. And we failed, and we couldn't do it. And we got in touch with the brain cancer folks and said, we don't know what's going on. We'll keep trying. And we said, well, maybe we missed something. Let's go back to the earlier papers and see if we can figure out what they must have tweaked to get something different. And in particular, in going back to the earlier papers, we looked more closely at the paper dealing with cisplatin. And then we looked at the heat maps real closely. And then we wrote up another note, which we sent to the editors of CCR and JCO saying, okay, they said this heat map was for cisplatin before. Now they're saying it's for temozolomide. One of these is wrong. We'll let you guys figure out which one it is. And a few weeks later, the editor of CCR sent us a note saying, well, the authors supplied the wrong figure. They will supply a new one. No need to publish your objection. And indeed, the authors a few weeks later supplied a new heat map and a new gene list, which didn't agree with each other or with the text in the paper. So in this particular case, they'd achieved the neat hat trick of having a paper nominally about temozolomide, drug A heat map for cisplatin, drug B, and a gene list that we still can identify for drug C. Neat, but still, that's what's going on. Now, with all of this as backdrop, this has taken years at this point. We're getting tired. Why do we care? Why should we keep going? Well, at about this point, in 2009, a friend of ours asked, by the way, are you aware that the PIs in question, who are based at Duke, have already started using this rule in clinical trials to determine what treatment patients get? And we said, no, we weren't aware of that. And we went to clinicaltrials.gov and we looked up what they were doing. And indeed, they were treating patients according to these rules. And in some of the trials, they were using signatures for drugs where, based on the data they'd posted, we were certain they had the sensitive and resistant labels reversed. We were not happy. We wrote up our various objections. As a single paper, we sent it off first to the editor of a prominent biological journal for informal comment. He got back to us and said, I'm sorry, your story is too negative. Can you fix that? And we said, no, we really don't think we can. So the next thing we did was we sent it off to the Annals of Applied Statistics because we figured most doctors would read it there too, so that's okay. And we got a different response there. The editor replied that evening saying, I like this paper. I'm going to handle its review myself, which we took as a good sign. And indeed, 2 weeks later, he sent us another email saying, we've simply decided to accept the paper as is. It's posted online now, which means I send more papers to that journal. I like that. But it also means that now that our paper was online and out in the public domain, we could talk about it to others, including members of the press. And indeed, just a few weeks after this appeared, the Cancer Letter ran a cover story saying biostatisticians at MD Anderson are alleging the potential for patient harm in clinical trials being run at Duke University. Oh, the horror. And we waited to see what was going on. And what was going on, something we didn't know, is that folks at the NCI read our paper and they looked at grant applications from the same group and they said, we're seeing the same mistakes being made here. So the NCI wrote to Duke and saying, we are concerned about patient safety at your institution. Will you reassure us? So Duke said, you know what, we're going to suspend enrollment in these trials. We're going to convene an outside panel of experts to review it to see if we should go forward. And we breathed a sigh of relief saying, okay, fine. We're not happy it went this far in the first place, but if they did, external review, that's fine by us. We'll wait and see what happened. And actually what happened next came out in the press first, because when Paul Goldberg of the Cancer Letter wrote about the story, he not only talked with us, he talked with the investigators at Duke. And he got a really interesting quote from one of them, the senior author, Joan Evans. And the quote that he got was that data was made available to us blinded. All we got was the gene expression data. We ran the predictions and sent it back to the EO and RTC investigators. Now, that's the data for the Lancet Oncology paper, which we had a lot of problems with. But if they got great predictions in a blinded validation, if they didn't know what the outcomes were and they still predicted it right, then they have something. So maybe we're missing something. So we said, is there something else that we can do? So the other thing that we did is we looked back at that paper, and this time we looked at the author list because the bioinformatics community isn't that large. And we looked at the author list and we said, oh, there's Mauro De Lorenzi from the Swiss Institute of Bioinformatics. We know Mauro. So we called Mauro and said, Mauro, how did they get such great results with a blinded validation? And Mauro's response was, who told you it was blinded? And we said, well, they did. And he said, let me send you the data that we sent them. So here is part of the table that they sent them, where they said, here is the name of the array, NPCR, EP, et cetera. The name of the array, this NP stands for no pathologic complete response. This one stands for pathologic complete response. In other words, every single array that they sent them was labeled with the outcome. They were never blinded. Furthermore, Mauro told us, by the way, if you could get the folks at Duke to tell you how they get these numbers, we'd like to know. They won't tell us. And this is their co-authors. So we said, okay, what's going on? So we asked Duke and got no comment. So we waited to see what would happen next. And what happened next was the investigators at Duke posted brand new data to the web to show how their signatures worked. The new data they were posting was dealing for cisplatin or pemetrexed. And this time they said, we got all the data we're using to validate this approach from this large data set, which is public. This public data set has 153 samples. We're just using 59, but we're using it to make great predictions. So we tried to say, let's download the entire data and check the correlations. So what you should see here, if the samples are what they say they are 59 red squares all falling on top of this blue line. We don't see that. As it happens in this plot, we only see 43 red squares. That means that for those 43 samples, we can perfectly match the numbers to a reported sample, and the sample isn't what they say it is. They got the name wrong. For these 16 samples over here, there's no red square. The reason there's no red square over there is because for those samples, they've already scrambled up the gene name so badly that these no longer correspond to anything biological anymore. But what I can tell you is that every one of their validation samples is wrong for drugs that they've been using in clinical trials for 2 years by this point. And we reported this to Duke and to the NCI to wait and see what happened next. And what happened next is that Duke announced they were going to restart the trials, because they had concluded their investigation, and their investigation's results had strengthened their confidence in this evolving approach to personalized cancer treatment. And we said, wait a minute, will you show us the data upon which this result or this finding is based? And Duke said no. Duke said, while the reviewers approved of our sharing the report with the NCI, we consider it a confidential document. A future paper will explain the methods. And we said, that's not how it's supposed to work. But if you look real closely at what they said, that actually gives us another in. Let's take a look at what they said. While the reviewers approved of our sharing the report with the NCI, the National Cancer Institute, is a federal agency, which means it is subject to the Freedom of Information Act. So a few months later, we got the report from the NCI pursuant to a FOIA request. And we said, this doesn't justify restarting trials. What the hell's going on? And Duke had no comment. And at this point, we were frustrated. We didn't know what else we could do. We couldn't see anything else in the data. We told people this was wrong. And we were trying to think of what would happen next. And what happened next is I got a phone call late at night in my office from a guy who said, hi, I'm a lawyer working in North Carolina. My client, who wishes to remain anonymous, thinks you should know some things about the investigators running this trial. I talked with this lawyer for a while, and then I said, here's the name of a reporter you should be talking with. And a few hours later, I got another call from Paul Goldberg of the Cancer Letter saying, I'm going to have fun. So why did Paul get to have fun? That's because a few weeks later, Paul got to run a story in the Cancer Letter saying that, oh, by the way, of the investigators on these clinical trials at Duke, one of them, the lead investigator, Anil Poti, used to claim on his CV and on grant applications that he was a Rhodes Scholar, but the Rhodes Foundation disagreed. He was put on administrative leave that day. This story came out on a Friday. I got a copy. I spammed all of my friends. I said, I know some of you talked about writing a letter. Now is politically the best time you will ever have. The following Monday, a letter from a whole bunch of prominent biostatisticians landed on the desk of Harold Varmus, the brand new head of the NCI, saying, please resuspend these trials until an independent organization can validate the science. Varmus got on the phone to Duke and he said, you're going to suspend these trials, aren't you? Duke said, yes. Varmus got on the phone to the Institute of Medicine and said, will you investigate this? They said, is stinky enough? Yes, we'll look at it. We'll start in December. Before we got to December, something else happened. Specifically in October, Joan Evans, the senior investigator, sent an email to his various co-authors saying, you know what, we might have to pull one of the papers. What paper might they have to pull? The one dealing with cisplatin and pemetrexed. Why might they have to pull it? Because on closer examination of their database, it appeared that 43 of their validation samples were mislabeled and they could not match the other 16, which is what we told them the previous year before they enrolled new patients. Shortly after that, Duke officially terminated the trials and announced that in retrospect, these trials should never have been run. After all of this, there have been a bunch of other developments. Patient lawsuits were filed. There was a misconduct investigation. Lots of papers retracted from such fly-by-night journals as the New England Journal, Nature Medicine, JAMA, you name it. They all got pulled. This hit the front page of the New York Times. It hit 60 Minutes. And actually, because I'm vain, I'm going to show you a brief clip from that 60 Minutes episode. This is Joseph Nevins. You're going to see him talking with Scott Pelley of CBS News. And we're going to click it up to make it big. Try it.
Dr. Nevins discovered that when the underlying data disproved Dr. Cody's theory, the data were changed.
It became clear that there was no explanation other than there was a manipulation of the data, a manipulation of somebody's credentials, and a manipulation of a lot of people's trust.
Manipulated data. These were not errors.
That's correct. It simply couldn't be random. It simply couldn't be inadvertent. It had to have been based on a desire to make something work.
Is it a close call, or is it abundantly clear that the data were fabricated?
Abundantly clear.
It is abundantly clear that the data were fabricated 5 years after clinical trials began. That's not the way things should work. Now, more broadly, for the students in the audience, what would you do? Something we found out in the course of the lawsuits was that there was a student at Duke who tried to raise these objections. Those objections didn't come to light until 5 years later because they were suppressed by folks at Duke. Now, some cautions and observations. This is a horror story. Most things don't go this bad all at once. We acknowledge that. But I'll tell you, we were able to recognize the individual mistakes, mislabeling of samples, because we've seen simple mistakes like these at MD Anderson. That's not because MD Anderson's a bad institution. MD Anderson's a great institution. It's because some of these mistakes are easy to make. It's easy to mix up sample labels and such, particularly if you're working in Excel. And what that makes us worried about is that because mistakes are easy to make, if they don't get fixed and you don't give us all the data, maybe because the journal won't let you have space, then it's often hard to spot those mistakes post facto, and that makes it hard to fix. Now, the thing is, this all crashed and burned in 2012, right about the same time you had Lee's paper and others coming out saying that not only can you have really bad case studies, but this is a pretty pervasive problem. It happens a lot. And that scared the NIH. They said, we don't want to do this. We want to get things working better. And in particular, why do we want to get things working better? Because here's what we think is going on. We think we're wasting half our budget. We think we're wasting close to $30 billion a year. What is it being wasted on? Agents and reference materials. This is, did you get the labels right? Study design. Did you avoid confounding? Data analysis and reporting. Did you tell us what you did? 90% of $30 billion for things that are easy to fix if you know that's what the problem is. So we think you should do it. Now, what have we and others suggested? We got asked about this to say, what do we need in order to reproduce a study? Well, in addition to the raw data, which we think is important at this point, we think we need the code, because we've got evidence now that simple written descriptions don't cut it anymore. The NCI got asked, various others asked, but quite honestly, there's a simpler way of stating what we want from a paper at the time of publication. At the time of publication, that's when journals have leverage. We want it all then because that's the time they're willing to commit and say, we believe that this is what's going on. So what does the NIH ask for? Well, here are some of the things the NIH asks for in grants now. Did you verify the identity of the cell lines that they're using? Are the labels correct? Did you discuss the experimental design? Did you avoid confounding? Are you going to share the data? Did you tell us what you did? They're not trying to make it impossible. it's doable. And furthermore, one of the things that we know it's doable because we've been doing it for several years. Here are some things that we did first. And for those of you who are curious, this is boiled down to a slide or two from a 3-hour short course I did for things. Here's videos. But basically, at MD Anderson, I looked at various others in the biostat group and said, could we screw these things up this badly? And we said, oh yeah, we can do it. So let's not do that. So what did we do first? We adopted a reproducibility mindset, which basically said, as we put together analyses and send these out, we're going to do so with the expectation that somebody else is going to run them and check them. What this meant in terms of our department was that we said, here's some ways of putting together code and data to make it easier to read. And before it leaves our department, a second person will run the report and confirm that the code gives what we say it does. I'd change things today because more open source tools are available that make it a lot easier to do. I'd use Markdown in our Markdown. This is an easy tool to learn. I'd use, and here are some real basic things. Within our group, we say, by the way, how do you name files? How do we name them in structure things so that everybody in the lab can look at it, find it, and understand what it means? How do we work publicly in Spirit so that we expect to put things on the web, we expect that people will check it and see what's going on? Furthermore, one thing I would do, and here's something for the audience, if I have to blow the whistle again, are there better rules for learning how to do it? Here's a good report from the Office of Ethics at the University of Illinois. So I recommend that. Now, I'm an optimist. I've seen things get much better over the past 10 years about how we report data. We're not there yet. We're still screwing up, and we've seen some of these with COVID papers, so it's still going on, but we're getting a lot better. And if you want to learn more, I'll give you lots of links. There's plenty of it. I'm happy to talk more because I ramble a lot, as you've probably figured out. But that's the links to where to learn more. And the last thing I have to do is I have to acknowledge that I didn't do this all myself. Here's a bunch of other folks who contributed to this. First among equals is Kevin Coombs. Between the two of us, we put a few thousand man-hours into assembling this reconstruction, which we really don't like doing, but that's what we had to do. So if you want to read more about it, go here. And the last thing I have to do is to thank you all for having put up with me for the past 50 minutes. That's what I got, folks.
Thank you, Keith. As always, it's such a great story and a great presenter. You do an excellent job of presenting it. So I don't know if you noticed, Keith's PDF has several links. All of these slides, as well as some other resources, will be made available to you on the GCC Rigor and Reproducibility site. So you'll be able to open those and click on those links if you want to actually access those resources. So I want to invite everybody to feel free to put your questions in the chat. We are officially into our break, so you're certainly welcome to go on your break, and we'll reconvene. Nope, that's okay. We actually went over into yours a little bit, knowing that we'd have this time for those that wanted to stick around and ask questions. So yeah, if you have any questions, please feel free to enter those into the chat. I'll ask you, Keith. You mentioned that you do think that the requirements for data have improved over the last few years. So do you have a sense of how this is actually impacting the degree to which we're seeing irreproducible work?
A few different ways that this has shown up. One is that we are seeing some journals impose new requirements about saying, have you checked this? Have you gone, can you provide us with, here's some checklists of what types of data that you've tracked. Actually, Nature requires this now, New England Journal requires this. We have also seen some initiatives partly guided by the NCI, partially funded by the Arnold Foundation, to conduct large-scale reproducibility studies to say, if we look at a survey of impactful papers, like in particular, there's a cancer reproducibility study where the group at the Arnold Foundation identified 50 high-impact publications, and they said, we're going to contract with new, smaller groups to replicate each of these studies. And once we conduct these studies, we're going to publish the findings. And they got together with the authors of, I want to say it was Peer J, where they, or eBiomedicine, sorry, I'm forgetting which one. And the journal said, if you conduct the reanalyses according to your experimental design, we'll publish them. positive or negative, will publish them. They got that pre-approval beforehand. What that has let them do is it's let them check and confirm what the reproducibility rates are, but it's also suggesting to the NCI and to other funding agencies that in cases where things look like they might be high impact, they're about to govern clinical trials or millions of dollars in funding, it might behoove the NCI and the other funding agencies to contract for a replication study first. Study often costs orders of magnitude less just to see does it work. This has also gone broader. It's impacted psychology and various others. I will point to Brian Nosek as a particular name, and there's the there are several new institutes that say, here's how you do good studies. And all this data is on the web, so it's free.
That's great. Okay. Got a question from Pavel. Thank you. That was awesome. My question is, it seems like you've had a lot of fight come from journals that publish this data. What benefit do the journals have for not enforcing these rules for reproducibility strictly? Seems like thorough proof of misconduct should be taken extremely seriously.
So I agree. And actually one of the disappointments we had was the response of the journals throughout the story. That said, a lot of the journals get a lot of critiques for several papers, particularly if they're high profiles. And many of the critiques are not always well thought out, is a way of politely putting it. This is particularly the case if you're dealing with smoking or if you're dealing with climate change. Some of the letters they get are really interesting to read. So as a result, they become really sensitive about this, particularly because the editors don't want to be in the position of judging the science. That's what they think the reviewers are doing. So if something comes in that's really strong, they're not sure what to do with it. They almost always send it to the initial authors for comment, etc. But one thing that I would do differently if I were to do this again today is that I would actually call the editors and say, I understand, for example, that you've decided not to publish my letter. That's fine, I'll live with that. But I want 5 minutes of your time to tell you why I think it's important and why I'm not a kook, so that they can give me or get that understanding from me. And actually, I'm telling you this because that's the feedback we got from the editor of the New England Journal and one of the editors at Nature, which is, why didn't you call us? I didn't call them because in statistics, that's not done. You don't call the editor of the papers you submitted, but it's done in biology. And actually, one of the things is what they'd gotten, because they talked with some of the initial authors said, these guys are on a vendetta and there's nothing to it. So actually, in those cases, they decided not to go there. To their credit, after all of this blew up, JCO, the editor at JCO, called us and said, you know, it's not our journal's policy to keep copies of papers we've decided not to publish, but it's come to our attention that a certain paper you submitted about 3 years ago was very likely correct. Would you be willing to resubmit? This was our paper dealing with cisplatin. So we bit our tongues and resubmitted. We submitted that paper. The paper on cisplatin was retracted the following week. So our publication has never been published and never will be. But they've gotten a bit better, but be prepared to also talk with people, because that's how conveying information is done differently within different disciplines, and you have to talk with the groups to say, how does it work here?
So I know that, you know, NIH has made recommendations for what journals should ask for and, you know, how they should, you know, try to address this. But at the end of the day, their recommendations, at least that's my understanding, is there actually any accountability, any incentive other than just, you know, reputation for these journals to actually have stricter requirements?
Well, in addition to the journals having requirements like this, I will also point out that some of the funding agencies and regulatory agencies also now have requirements. So for example, for clinical trials that the NCI funds, these are now requirements. You've got to supply this data. You've actually got to make some of it public, or we won't give you money. So it's not just the journals asking for it. But another thing that is happening in journals, and this is something I am seeing more in journals outside of oncology, but certainly in applied math and computer science, is that the speed of publication and analysis is ramping up because people are able to see the code and they're able to build on previous studies more quickly. So that has increased the rate of development by lots of people seeing it. A brief note about COVID, because this is somewhat related. Because people are talking about high impact publications a lot, one of the things that came up a few weeks ago was that there was a high profile study from a group called Surgisphere, which said we figured out how well hydroxychloroquine works as a treatment for COVID. And they claimed great results. And a bunch of statisticians who were chatting about COVID online started looking at it and they said, this makes no sense. In particular, they're reporting more deaths in Australia than there were cases. That doesn't make sense. And they're reporting, they chatted with each other. And the thing is, they identified several errors like that. And they had 20 or 30 folks identifying all at the same time. And they were all sharing data, which built up a critical mass. Because one of the things about objecting to a paper is that you're always worried that maybe I missed something. So you're very hesitant to critique something publicly. But if you've got a group of 20 or 30 independent folks and you're all saying, no, this is screwed up, you feel a good deal more comfortable putting together a communal letter saying, by the way, guys, this is wrong. Say something about it.
Well, Sharice would like to ask, how did you manage to fund your inquiry? Did you have to do this on your own dime?
That's a good question, because as you probably gathered, this does take a substantial amount of time. This forensic bioinformatics is not an approach that will scale to deal with the problem. The thing that helped for us was that the underlying papers were so high profile. And the other thing that helped was that Kevin Coombs and I were senior bioinformaticians, and that meant that part of our salary, our day-to-day grant salary, was coming from large grants at MD Anderson, like the ovarian spore and the lung spore and the melanoma spore. These are all groups that say, we got a lot of money to look at this type of disease. And since some of the PIs of those grants were coming to us saying, we want to use this if it works, we got to turn around and say to the PIs of those grants, you know what? For the next few months, we're going to charge the time of yours that we have allocated for your grant. We're going to charge it to get this to work. And they were okay with that because if it worked, this was clinically really important and they could use it. So we had to pitch it. We had to pitch it that way, but that's how we sold it initially. Later on, after the clinical trials issue came up, that we couldn't really build to the other ones. But what happened there is the fact that, well, we're senior faculty at MD Anderson. MD Anderson allows us some protected time. As it happens, I had 30% time that I didn't have to have grant funded. And this became my protected time for the next year or two because I thought it was important and I didn't see a way to directly fund it otherwise. So.
All right. Well, thank you so much, Keith. And thank you. You've got several thank yous in the chat. Great talk as always. Everybody enjoy a short break. We will start promptly at noon. Okay, I want to go ahead and welcome everyone back. It's time for our working lunch experimental design panel discussion. First, I want to just kind of introduce our panelists today, our Robia Potler from Baylor, who's an expert in small animal models, Dan Harrington from UT Health, who's an expert in cell culture and antibodies. By the way, as I say these, I recognize that they're also expert in other things too, but they're going to be talking specifically about these things today. Natasha Kirienko in antibacterial research and discovery, and Kurt Zhang from IBT in biostatistics and bioinformatics. So I'm going to invite all of our panelists to just introduce themselves briefly in about a minute and a half or less. We'll start with Robia. You want to go first?
Okay, hi. Thanks, Suzanne. My name is Robia Potler. I'm an associate professor in the Department of Molecular Physiology and Biophysics at Baylor. I've been working with small animal models for many, many, many years. Actually, first learning how to do my first small animal surgeries when I was 14 years old. So it has definitely been a while. Also, my expertise is in small animal MRI, and I've been participating in this workshop with Suzanne for a while now, and I think it's terrific.
Thank you. Dan.
Hi, everyone. Good afternoon. I'm Dan Harrington. I'm an assistant professor over at UT Health. I've been doing cell culture since graduate school and particularly in 3D cultures. And what has really emerged over the years has been a focus in knowing what you're using, your quality of your reagents, where your reagents can be something chemical or it can be your cell lines or your antibodies, and the recognition that those biologics need validation has really become important as folks finish an experiment and they get strange results and they realize they were using HeLa cells the whole time. So that's part of what we want to talk about today.
Thanks, Dan. Natasha Kirienko from Rice. We have Natasha on the line.
Hello. My research is a little bit diverse. So I study infections. I do lots of microbiology with multi-drug resistant bacteria, specifically Pseudomonas aeruginosa. But I also work with cancers, with leukemias. And in both cases, we look for how do we treat the pathology, and we often work with drug combinations. So in both treating cancer and in treating bacterial infection, we look how to find drugs that will synergize with each other and how assess what effect they have and how do we know that that's reproducible.
Thanks, Natasha. Kurt Jang, are you on the phone?
Hi, yeah, I'm Kurt Zhang. I'm from IBT Institute of Biosciences and Technology. That's an institute in Texas A&M University. My research is mostly focused on computational biology, so that's including biostatistics, bioinformatics, most for genomics data. And my lab also do some white lab stuff, like use a small animal like a mouse. So work on a bunch of projects like heart development, diabetes, obesity studies. Thank you.
Great, thank you, Kurt. Okay, so I'm going to invite Robia to share some slides. We're going to go through some of the cases and questions that you submitted. We're going to invite our panelists to chime in. I want to say we recognize that while there's a lot of expertise on our panel, you also in the audience attending this workshop have even different expertise. I want to invite everybody to share your thoughts in the chat, and I will share them as I can. We're also going to be providing the transcript of this chat in the resources in the file for today's workshop, so you can go back and look at it. Also invite the panelists to continue to look at the chat and answer questions if you can. And we'll also have some polls here. So Dawn, who is, we're gonna go back to the first slide. Okay, so Dawn, this first question, we're gonna do a panel before we a poll before we discuss. So Dawn's gonna share that, and if you'll just answer that, and then we'll kind of see what everybody thinks about this one before we discuss. So if you could take a moment to quickly read through this and answer what you think the best answer would be. And Dawn, once it looks like most people have answered, we can go ahead and share those results. We'll take about 10 more seconds, and then we'll go ahead and share. So how should one respond to an assertion that their study is not reproducible? We had a couple for A and B. The majority of you think that you should discuss the reasons for the assertion, see if there's an obvious solution. Some say discuss with your PI, offer to repeat the experiment with the person who made the assertion. Looks like we have one person who thinks you can ignore it if your PI didn't say it, and maybe one person that if you think you did everything correctly, you can just assume they're wrong. So thank you, Dawn. You can close that, and we'll turn that over to our panel.
Okay. I guess I'll field it. I think that C is the right answer, and it was good to see that the majority of the people in this workshop seem to agree with that. However, I can also appreciate that if you're a student, you're first going to want to go discuss with your PI, and that's certainly not the wrong thing to do. But I think the follow-up should definitely be to not ignore it, but to definitely address it. And the other thing that I wanted to mention, just thinking about how these things can happen, it's also an opportunity to really practice your soft skills, because sometimes when people come to you with these assertions, they aren't maybe always friendly about that. It might feel accusatory, it might come off as aggressive. And what I would really encourage individuals to do, if should they ever find themselves in this situation, is to remain calm and always be professional and polite. We're always here to get to the truth. That's what we're seeking as scientists, and just get to the bottom, and again, see if there's an obvious solution and find out what may or may not have happened, because this does happen. And oftentimes, as you'll see in some of the other questions, or as Dan kind of alluded to, sometimes it just could be like mix-ups in genotypes, like the difference between very slight differences between strains, like C57 black 6 Ns versus Js. Or it could be perhaps the diet that, for example, in mouse models, it was slightly different. And I've certainly run into those issues myself. But I'll let the rest of the panel also address this question too. So,
Dan, you have anything to add?
I think that's a great response. I really appreciate the idea of the soft skills, because that certainly can happen, and it's a good opportunity to work together as a team and try to find out maybe something people have missed.
Mm-hmm.
I think you nailed it there.
Natasha?
And I think it's also important to discuss it, especially if it's for a student, to discuss it with PI, because sometimes PIs have better understanding of what's important when. And there are, for example, certain protocols where osmolarity or pH are critical. And I've seen cases where lab couldn't reproduce something because their pH meter wasn't working. But student may just be going through protocol and doing everything correctly without understanding which steps are important, which not. So if having PIs involved helps.
Yep, agree. Kurt, anything to add to that? I think you're muted, Kurt. I can't hear you.
Yeah, I agree with everyone. I think many times, I mean, we should not assume the assertion is right or wrong. So many times it's possibly just happened the condition is not specified accurately or enough. So give it enough detail to reproduce the results. So as Dr. Porter said, so like the genotype, you didn't specify a particular genotype. Possibly that's the reason you couldn't reproduce, and you use different cells to reproduce. Possibly that needs different results. So yeah, if everything is really specified and we suppose a different lab can easily follow the procedure to do it, and cannot do it, then possibly there's something we need to consider about the science or mechanism behind it.
Sure. Thank you very much. So I don't see any additional questions in the chat on that one. So Roby, we can go to the next one.
Okay.
So do you really believe it? What are best practices for ensuring reproducibility standards have been followed in publications? So this is just a discussion, and would certainly love to hear thoughts from the audience as well. I mean, when you're looking at a paper, when do you get excited and when do you say, wait a minute, that looks good, but I really don't think they did the right experiments. I'm not convinced that I need to waste my time exploring this. So how about Dan, let's start with you this time.
Sure. You know, I think it's from two perspectives. One is the writer and one is the reader. I just returned a review back day before yesterday saying to comply with NIH Rigor and reproducibility, please indicate sex as a biological variable in the cells that you extracted from this particular tissue. You know, we're asked to follow those guidelines. When I'm reading a paper and trying to see if it's been written to this standard, I just think, if I blanked myself, could I follow this set of methods and reproduce the experiment? Could I do it? Or could I offer this to an early student in the lab and see if he or she could reproduce it? And if not, that's the moment, you know, and that is maybe listing the exact clone numbers that your antibodies were. So that it doesn't just say that it was a vimentin antibody from Sigma, where there could be 8 different versions. Similarly, if you are using a cell line indicating that it had been checked, you know, and how do you check it? You do an STR comparison to make sure that the cell lines you have, the cell lines you used, are the ones that have a published signature, to make sure that, as I said, it's not HeLa cells. We even have a story in the lab of somebody using canine cells instead of human cells. They call that the Snoopy experiment. Those kinds of things, I think, on the writing side.
So Sanjana says that this happens most often to me in immunoblots presented in papers. It's exciting to see bands, but can never always reproduce it with the same antibody used in the paper. And I know this is an area that you kind of specialize in, Dan.
You know, it is a challenge, and the Western blot desert is where people often find themselves, right? Trying to reproduce those beautiful bands. And so it can be a lot of things, you know. It can be, how did they block? What did they block with? How did they, you know, there's so many parameters in a Western. And I think you learn something as you go about good ways to technically perform it, but then it is those details of what was the dilution, what antibody did they use for all these steps, and being thoughtful as a writer to include that. Just think of the person who's coming after you, pay it forward to them so that they can reproduce your work. If you're stuck, it's never against the rules to write the authors and say, I'm having some trouble. Do you think we could have a Zoom meeting, and maybe you can give me a couple more details that were maybe unclear to me from the paper?
I have a comment that we need to be more supportive of people having negative data. Recently, I was reviewing a paper where they were talking about activation of MAPK cascade. So I asked, why wouldn't you use anti-phospho-MAPK antibody and measure active level of the protein? And the response was, we got the antibody, we tried multiple times, we can't, because this antibody was cited, and they said, we just couldn't reproduce, we're getting multiple bands, we can't get the results that authors got in the paper when they published it. And as a reviewer, I said that I appreciate your effort, just please tone down that based on other indirect methods you expect pathway to be activated. And don't worry that you not being able to reproduce somebody else's data can be held against you.
Any other thoughts?
Yeah, I'd like to jump in here. I wanted to note too that oftentimes you'll see, and this is becoming a more and more prevalent trend in a lot of the journals, is that they will ask you to complete a key resources table. And in doing so, you just have to list all the resources that you used, you know, like maybe what group number or what antibody, specific antibody information that you have, the mouse information, and that's included as part of your submission for your manuscript. So Suzanne, I think, included a table template from the journal Cell, But what I am recommending to people in my lab, and I highly recommend all of you consider this as well, as you're going along, just complete the table, have your dates, be able to cross-reference it to your lab notebooks or even put it in your lab notebook. So you know specifically what was used, and that would also help you troubleshoot in the future should you encounter problems. So it might even be like a certain lot number of the antibodies might have been ineffective, and that's one way of tracking it.
Yeah. Anything to add, Kurt?
Yeah, I agree with everyone, but I think this is a rather broad question. So that's almost counter everything we already learned today. So besides the authentication of the reagents, also it's concerned about your experiment designs and your data analysis. Have you controlled all the biological variables?
Mm-hmm.
So yeah, so that's basically everything we need to consider into.
Yeah, there's a lot even beyond the reagents for sure.
Yep.
Okay, Robia, let's go to the next one. Okay, so we've got a poll here asking, when is it okay to exclude outliers? Dawn, you want to go ahead and share that? Okay, we'll take about 5 more seconds. All right, let's see what the audience thinks.
Hey.
All right. How about Kurt? Why don't you start this one?
All right. Well, this is difficult one. Okay. So yeah, look at this 4 answers. Certainly B stand out. So, but in general, I mean, if I'm as a statistician, I will recommend you don't exclude anything unless you're completely sure. So when I say completely sure, it means really think that's the outlier come from the biological variables, like the mouse is sick. So you know it's sick and its behavior different from ours, then yeah, we have that biological evidence, then we can include it. Otherwise, yeah, normally, yeah, generally, I would recommend you include. I mean, even if it's an outlier, because that's just one part of your data, and that's possibly when you run many times, it's always there's something out there, and we'll never be able to control. So, but yeah, in terms of statistics, so certainly there's some guideline. I mean, for statistics to determine which is outlier. So sometimes we say, okay, if it's 3 times of the standard deviation, then we can determine as outlier. But yeah, even for those guidelines, still, I think you need to be extremely cautious and yeah, and the biological knowledge is most important to determine other than statistical evidence.
We always
I agree with Kurt that we need to be very careful excluding things because we don't know biological variability of the process. So it's better not to exclude outliers.
We always had a rule that unless you had specifically noted in your notebook when you did the experiment that something was messed up in this animal or this well, you know, there was some kind of mistake with an injection or a pipette or something that was identified in the beginning as a mistake, you could not exclude it.
Yep.
But for example, we work with lots of drugs that are very freeze-thaw sensitive. We can include 2 microliters for single experiment. And sometimes our freezer fluctuates. So we always, when we look for drug combination, we always include single drug control for which we know how it should perform because we did it hundreds of times. And we have before we do experiment, before we started the series of experiments, we have an understanding that if single drug has LD50 of certain concentration and there is no killing even at 2x LD50, then the drug just went bad and we have to throw out whole experiment.
Yep.
Mhm.
These criteria were selected even before we went to the experimentation, and we're sticking to them. And that helps us to only focus on the data that may be variable. We have different degree of synergy and interactions, but at least we know we're not introducing artifact due to not working drugs. The more controls you can build into your experiment, the better your outcome would be.
Anything else that if the panelists would like to add?
Nope.
All right, well, we can move on to the next one. So here we have a case study, and this is one where we'll just invite our panelists to weigh in. But again, I really invite you to enter your thoughts into the chat. An invited paper, due dates approaching, but one of the figures could not be updated in time because the analysis took too long. Though the pattern using the new data was expected to be similar to the old one, and it actually did indeed to be the case afterwards, there was a debate between the authors, and one suggested including the old figure at first and then replacing it during revision after the experiment had been done. The other author worried that reviewers would not accept the exchange of figures later without a convincing reason. Asking for an extension could result in losing the invitation. So what's the best way to move forward and to convince the other authors?
It started that as I recently had a lot of experience being a guest editor for invited topic and writing an couple invited reviews. So especially people generally tend to be really understanding with deadlines. If somebody plans to release a topic, the goal of the editor have as many articles there as possible because it will attract more readers. And people are willing to increase deadlines. Now, during COVID time, if the increase in deadlines is almost automatic, instead of maybe 2 weeks, 1 month, now you can get up to 2-3 months extension of deadlines. It doesn't hurt to ask, especially explaining that you're waiting for the last figure. And recently, as authors, we were also late in submitting to a special topic, and we explained situation to editors in advance before the deadline. We said, we have this data, but we're waiting for next batch of molecules to complete experiments. Would you consider waiting? And editors feel appreciative that we were proactive and said, sure, it's fine. Rather than worrying that invitation may be rescinded, try contacting editors and asking what do they think. And likely they'll say, submit later, not a big deal. You'll save all the time of worrying.
So in the event that you do that and they say no, what do you think about just submitting the old figure and planning to trade it out later?
I personally would be too scared to do that.
I wouldn't do it.
All right. Any other thoughts from the panel? Okay, let's move to the next one. During the pandemic, we've seen seen new therapies fast-tracked to clinical trials, possibly with lower standards of rigor and reproducibility. How do scientists and physicians weigh the ethics of fast-tracking vaccines and therapies when faced with a pandemic? Let's see, who wants to start with that one?
I'll riff off of what Natasha said earlier, that there's a lot of value in establishing in advance what your guidelines are going to be. You know, when you set up your own criteria for success or failure, then it's clear in advance. You know, and you stick to it. You know, that is the ethical question. It is always a challenge because there's always, in this case, there's always one patient who would benefit if the result was available a week earlier. And all you can do is your very best to be as efficient as you can, as quick as you can, but also rigorous and certain to the best of your ability. That's my feeling.
I don't know if you remember Keith talking about the Surgisphere study on hydroxychloroquine, but I think it's, you know, it's encouraging that because this is such a high profile thing that there are maybe more watchdogs out there watching, and trying to, really hold people accountable. And, if you've got more people, like he said, looking and they're all finding the same mistakes, but that begs the question of, some of these things that aren't always published before they move forward. So I think it's a good question. Anybody else have something to add to this?
So I have just a case study, COVID related. There was a paper that about COVID saying that inactivation at relatively low temperature, 55 Celsius, is sufficient to inactivate COVID. And that paper was in bioarchive for months. And there were other papers who were saying that that's not sufficient and you need higher temperature. And these papers were actually published. So the thing is, you need to be really careful especially when you're looking into something like bioarchive that's not peer reviewed. On one hand, you have access to most cutting edge advanced science. On the other hand, though, you have less chances of knowing how rigorously that was done. So always, especially with COVID related research, because there are lots of people doing relatively similar experiments, there are likely more than one publication talking about something. So you'll be able to do it by cross-referencing. And if you'll see a couple studies that agrees on something, that likely is on the right track. But if you'll see contradicting evidence, that's where more scrutiny will be needed.
Yeah, just one more comment. I mean, when we do this kind of time-sensitive publication, so there's a relaxed criteria, for example, then I think the author should, and also both the author and the editor should put a note discussing something to say, okay, we actually, because of time constraint, so we relax some criteria and we have the detail, what kind and what's the risk to interpret our current data.
Okay.
I'll
go ahead, Dan.
I'll add to that and I'll just say, this was, I think, still memorable to folks regarding COVID. A paper came out demonstrating a laser kind of technique, a light scattering technique for assessing mask materials. It got a lot of press, and I think it at the time was hugely influential in kind of swaying people's opinions based on how quickly the broader kind of press picked up this article. But if you dig into it, it really wasn't meant to be an article that rigorously assessed materials to tell you one is better than the other. It was a demonstration of a method that could be used. But yet it was so influential in changing people's opinions. I know that my daughter's school completely changed their mask recommendations based on this one paper, which had a purpose that was not to make that recommendation. So it's up to us to be really to question things, back to that, I think that earliest question, it's worthwhile to ask questions and decide if a study makes sense and is appropriate for an intended use.
Thanks, Dan. Okay, Robia, we'll move to the next one. Okay, so we have another poll question here. Let's take a few seconds to answer this one. Okay, we'll take just about 3 or 4 more seconds and then see what the audience has to say. Okay, Dawn? I'll say this one's kind of not fair because it only allowed you to pick one, and this one actually had more than one answer. So Robia, I know you're chomping at the bit.
I am totally chomping at the bit on this one.
Okay.
As Suzanne said, there are multiple correct answers to this. So what has been recently implemented in the last few years is instead of scientific rigor, or I think that's what it was called, scientific rigor, it's now called rigor of prior research. And rigor of prior research is actually, or rigor of research is split into two segments now. It's rigor of prior research and the rigor of the proposed research. Both of these are scoreable. So the rigor of prior research is in the significance section. And what you need to do is you need to outline what has been done up to this point of what you're proposing to do, and basically gives the impetus for why you're proposing what you hope to do and what holes you propose to fill and how you will advance the scientific knowledge. The rigor of the proposed research is in the approach section, and that just assures everybody that you've carefully thought about the methods that you are proposing to use to address your research question, and it is indeed a rigorous process. That is also scoreable. Another scoreable element in grant applications is consideration of relevant biological variables such as sex. So again, that's going to be in the approach section, again, scoreable. And then the last component is a completion of a specific attachment, which is the authentication of key biological variables and chemical resources. So this is actually not a scoreable element in reviewing grant applications, but it should be included. If you do miss it, I mean, the program officer can ask PIs to revise it if they are being considered for funding. So what Suzanne has shared, I think on one of the drives for everybody who's a participant in this workshop, is a slide that I'm given as a reviewer that actually states, you know, what are reviewers looking for in each of these components, and which are scoreable and which are not scoreable. But again, I would really encourage people to even spell out in their grant applications, and this isn't just the R series grants, this also applies to fellowship grants. I would bold and highlight the rigor of prior research and rigor of proposed research. How do you specifically address these? Because that way you're just specifically leading the reviewers to letting them know that you have addressed each of these components that you are supposed to, and then you can spell it out for them and kind of make their jobs a little bit easier, which of course reviewers appreciate. But I guess I'll get off my soapbox then. But I mean, we can go delve more into the specifics of what is required in each of these as we go through this panel discussion. But I think I should probably let the other panelists chime in here too.
Well, I am going to throw out, I mean, you know, when you're trying to assess the rigor of the prior research, I mean, you looked at their papers. I mean, give an example. What does that look like? What do reviewers want to see?
What we want to see is that you've basically built up what have you done and what have others in the field done. So if you have not done a comprehensive job describing what's been done in your specific field or your specific research question, you're going to get dinged on it. The other thing that you can point out is, let's say that there was a paper that was similar to what you're proposing to do, but it was not a rigorous design. So it's it's questionable within the research community. You can also point that out nicely. You know, it's not you're not supposed to trash people in your significance section, but you have to be objective about it. It's just like, what are some of the holes in the what's been done so far? You could say, well, there is some promising work done by Dr. So-and-so's lab, but there was perhaps a lack of rigor because of XYZ. And we propose to address this and advance the scientific knowledge by doing QRS. So that would be like one example.
Okay. Other panelists?
I have a generic comment that over recent years, there is more emphasis on doing mentoring and doing research rigorously and reproducibly. So there's a recommendation, even in your personal statement, to talk about how you being a mentor, how you're not only doing rigorous and reproducible research, but how you're training your mentees to do the same. And actually, I have it in my personal statement, and I had it positively commented a couple of times. And also, and evidence from my graduate student recent application, that student made all the check marks for all the rigor and reproducibility training required and applied for 2-year grant without proposing any more training. And reviewer's comment was that rigor and reproducibility is not just a check mark, it's continuous process. So you need to show that you're doing it constantly. So keep that in mind.
Tasha. Anybody else before we move to the next one?
You know,
I
I'll
just-
build the
go ahead.
example and just say that ways that you might improve would be to say, well, this great landmark paper had 30 patients, and now because we're in the med center, we have access to a database of 400 patients, and that improves it. Or this database, this study was restricted to you know, white patients only, and we have a diverse population, and that will increase, you know, some elements. So there are ways you can, I think, as was stated, kind of without bashing previous work, just demonstrate that you're going to be building upon it with the new resources and abilities that you have.
Okay, let's move to the next one.
Okay.
I think it builds on this one. So we've talked about these different things. How important is it, this rigor and reproducibility section on your NIH grant proposal? Okay, about 3 more seconds. Okay, Dawn, let's see the results. So some think it's make or break, and some know that it's a scored section. Some think it's considered a tiebreaker. So panel, let's talk about these different suggestions. Okay, Natasha.
Of course, its components would be score, but I think depending on the PI, if it's somebody with established lab who is known in the field, known for rigor and reproducibility, maybe a little bit more slack from your reviewers. But if you're a junior faculty, you don't have that much of a track record, that I think it absolutely can make or break your proposal if senior colleagues think you're not doing things reproducibly. So even as an established PI, it's very important to have it in your grant, but then you can get away with a little bit less than if you're just starting and you need to establish your credibility.
Yeah, I'm going to jump in here too, because when we do the trainings and I have to do this repeatedly, so I review every single review cycle, sometimes twice, and they do ask us to go through a training on this. We are specifically told not to give breaks to senior faculty if they don't appropriately address the rigor and reproducibility. It's just like you evaluate the grant that's in front of you, and you review it based upon its merits, you know, the document that you have in front of you. So even if it's somebody who's very well known, you say, well, I'm sure they'll figure it out. You can't do that. I think the answers are both A and D. It is a scored section, and it is absolute make or break, because if you don't have it, you're going to get a bad score, which can then break your grant. So I think A and D are definitely the right answers right here. Yeah, I think, well, here's another thing that I wanted to bring up. So this was actually, I'm about to review actually in a couple of weeks here for a specific study section. And the SRO brought up a point that one of the reviewers asked her, is just like, well, does a COVID contingency plan fall under rigor and reproducibility? You know, do we ding people who don't include this section within their grants? And the answer at this point is no, we do not. It is not required by NIH, but it could be asked for if you are being considered for funding in a just-in-time document to include a COVID contingency plan by your program officer. So I just wanted to clarify that, because it does fall under kind of rigor and reproducibility in these really interesting times, but that's not required. And I'll let Kurt and Dan jump in here too if they would like to.
I'll just add one thing, that grant rules have been, and guidelines have been changing rapidly. And the portions that we were just talking about earlier called premise, and not that long ago, you know, maybe I think 2 years ago, and then we're adjusted, you know, and those of us who write routinely, you know, or even folks who just maybe haven't done it in quite a little bit, it is worth checking and rereading carefully, because these things change annually, it seems. You know, for tweaking these elements to make the process stronger, it just requires keeping an eye on it. Because I remember when sex as a biological variable first became an important topic, I remember seeing the first grant that came back with a big ding because of missing that point.
Hey, Dan, can I ask you something?
Yeah.
Or I want to ask the panel too, or just the whole audience. How do you find out about these updates? Because for me, it's through the grapevine or it's through sitting on study section. And then that way I'm able to capture that information and put it into my own grants. But then you see a lot of people getting dinged for it when they have an otherwise really strong proposal. So I'm wondering if this is maybe something we should consider suggesting to Suzanne and the Gulf Coast Consortia. Maybe there could be like a grant update small section. I don't, you know, just like so people can go to that or be advised of it. Just when these updates do happen, I think it could help a ton of people.
Such a good point. Yeah, I think I tend to find, I've not been on any NIH study sections yet, only for other agencies. And so I don't find out through there. And there is a little bit of grapevine, but I think there still are a number of listservs that you can sign up through email to receive updates from NIH as one agency. And when you're joining another new agency, there's definitely some reading to do. You know, like preparing an NSF application is just substantially different, and it takes a while to understand that culture and to get some information. And you may be an experienced writer, you know, you may be experienced from your F award, you may be experienced from your first R01, you may be, you know, and it may be a couple of years since you've submitted something. You want to check with some folks to give them time to pre-read your grant with and for you, and maybe they comment on things that they've seen.
Yeah, I just wonder too about like individuals who are fervently writing and maybe missing some of this information, and that definitely does happen.
No doubt about it. All right, let's go ahead and move on to the next one. Okay, another poll. Okay, do another 3 seconds or so. Okay, Dawn, let's see the results. So wow, we're almost split between both results should be reported, and to avoid rejection by editorial review, you should perform more experiments before publishing. Okay, Kurt, you want to start off on this one?
Sure. Well, when I read this question, I mean, I will quickly think about the statistical point of view, but yeah, sounds like this question is more for the like a selection bias. People tend to select the positive results. Yeah. So in that sense, sounds like the D is correct. So because at this stage, I mean, you have two conflict results. So you possibly need to do more experiments. But yeah, but in terms of reporting, so if you stop there, then I think A is right also. So you need to report both results. But as a statistician, I would think more about the approach. So I would think, okay, when you do an experiment, I mean, say first time you got non-significant results, are you going to do another experiment? Or the first time you got significant results, possibly you just report, yeah, stop there and report. So what's the right procedure on that? So yeah, if I think that way, this question become very complicated. So I think there's another question for a few questions later. So maybe I can suggest yeah, more detail over there.
Okay. Anybody else want to add anything to this one? I will say before we move on to the next one, we did have a comment from the audience regarding the last slides that they get regular updates from NIH regarding changes, like we were talking about ad nauseum, and it's definitely a challenge to read them and keep up. So
Well, I'm just wondering if just like a quick bullet point type of thing that's easy to read would be useful.
yeah, certainly something we can think about.
Yeah.
All right, let's go ahead and move to the next one.
Oh.
So a discussion question. Like to hear from the audience on this one as well. How can you enhance interlab reproducibility in animal studies, especially when gut microbiota have such a huge impact on host physiology? Roby, you sound like this one scares you. You're our animal.
Oh my God. Well, only because we were collaborating with a group where we were trying to help them image intussusceptions. And the first round of mice, it was gorgeous. The mice who were exposed to a certain environment or drug, or it was, I forgot what challenge it was, but they actually readily developed intussusceptions. But then the mice were moved to a different animal facility, and unbeknownst to us, were actually given a different type of food. And it took us over a year to figure out what the heck was going on. And so when things like that happen, you can really, really lose some time, and you can just truly drive yourself absolutely insane. And I think this is a really good question because animals are really, really sensitive. So even just like small changes in environment can really change or alter the gut microbiota and a lot of their physiology. So I think really recording like light dark cycles, maybe even just like the humidity, the temperature, how often cages are changed, all of that can affect animals, not just gut microbiota, it can affect the matings. I think it was a few years ago, there was a change in the way Baylor College of Medicine, the type of water they were giving to the mice, and that screwed up a lot of studies too. And there was a lot of problems, which I think have since then been rectified, but it did kind of throw a wrench in a lot of studies. But I'm just going to end by saying this is an absolutely huge issue, and the best way to try to deal with it is to try to make the environment and the feeding as reproducible and as close as possible to the studies that are reported. Hopefully when you're conducting such studies, people are very, very careful in reporting all of the details. But it can be a nightmare.
Anything else?
I just want to add something. So yeah, besides that, so certainly all the conditions we should be well controlled. But in terms of data analysis, so we need those things we call is batch effect, right? Between different lab, different animal facility. I mean, there's something you can never be controlled. So always show some batch effect, some difference between those labs. So one thing we can take into consideration in the data analysis is we think about what's endpoints, what's the data is possibly will not change. For example, in the microbiota, possibly we're looking for alpha diversity, beta diversity. Hopefully those things will not change. Even the individual species, bacterial species, their load, their count of change. But the diversity, hopefully relatively stable. So we're looking into those rather, more stable properties and do the data analysis. And also in terms of data analysis, there's many ways we can control the, can normalize and remove the batch effects. But yes, sometimes it works, sometimes it doesn't work. And yes, but it's possibly always still some progress, some improvement.
Okay, we'll move to the next one. Oh, well, we do have a comment from the audience. Sanjana says, probably best to allocate one room to breed the animals from the different labs so as to avoid the issues in environmental and other factors. So I think everybody's in agreement.
Yeah.
Okay, so new poll question. At what point are key resources expected to be authenticated? Okay, take a couple more seconds here. Okay, Don, show us the results. So the majority think once you get it, once you acquire the resource, and several say definitely before publishing. A few, once you have 3 or more reproducible experiments. So panel, what do you think?
Well, I think it's a little bit of a level of risk to some degree. So some people might write in their authentication document that they rely on the vendor to provide the authentication for a new reagent, so that your toluene is 99 point something percent, or that your cells are validated by STR for being that specific line, or etc. That these things come with a certificate of authentication, and that may be a risk that a group is willing to take and say we take them for what they're given as. But typically, I mean, I think I'd put E before you do the experiment, because it's certainly much harder to do it after the experiment, after you've done all the work and it's a whole summer, or a whole lot of work from somebody, and something's not right, or things aren't adding up, or the journal says what's your authentication, and you realize that it didn't add up, and you wish you'd done it from the very start. So I guess that issue with D is just, what's your lab's criteria for saying that a resource is, correct?
Dan, along those same lines, we had one of the assessment questions that asked about this regarding your cell line. What's the best way to confirm the identity of your cell line? What do you think about that?
Well, that STR method that I mentioned is one way that really gives you a definitive fingerprint. There's a few variants on how people are authenticating their lines that way, but today. But if you know that Catherine in the lab was really meticulous, and you grab one of her vials instead of Peter's vials because he was always a mess. No, right? That's not a rigorous authentication method. Anytime you dig into the smokiness of the liquid nitrogen dewar and you pull out a vial, you have to wonder what's in there. Was it infected that day? Did that person accidentally mislabel? Did it go into the wrong vial? And if you're going to be spending time with those cells for the next months or a year, you want to know from the start that they're the right ones. And it's easy to get that done locally. It's not super expensive. It's usually under $100 for a line. And
are those
cores?
Yeah. Individual institutions? Okay.
Yeah, I think there is a core at MD Anderson. I think you can also send it out to a lot of different places around the country. And
okay.
You wait a week to get that result back, maybe a week and a half, but that's definitely worth it.
Yeah. Okay.
I have a comment on relying on core. We use C. elegans quite a bit in our research. And there have been cases when we'll obtain a strain from an occult consortium, and instead of homozygous mutant, it would be heterozygous. The first time it happened to me when I was a grad student just starting and didn't realize that I need to amplify it right away. Luckily, we were able to figure it out before doing much work with it, and then from heterozygous got homozygous mutants. But now, if we're receiving a new mutations, we always confirm that this is complete deletion, and then we freeze a lot of vials, because random mutagenesis happens. Same with cell lines. After you use it for a year, you don't know what else is happening in that cell line. So after a certain amount of time, a year for worms and about half a year for cell lines, we'll just take a new tube from the freezer and switching to the new tube. That was helpful so far.
Yeah. I completely agree with what she just said. And it not only applies to cell lines, but also to reagents. For example, we work with nanoparticles in our research, and they don't last forever. Their efficacy does not last forever. So I think kind of like what Dan was saying, just perhaps even before the experiment, just to make sure everything is in proper working order, it's imperative to do that.
So I think we have time for one more. Let's move to the next one. Using the same mouse model, my lab is unable to reproduce the data published by a collaborator. Information is crucial, especially to patient populations. However, negative data are usually considered not attractive enough to be published. What should our lab do with these data? Important for the patient population. What do you do?
You dig deep. You find out what may or may not have been the same. I think there's a lot of background information that needs to be done. Really a lot of comparing notes. I think I also agree with what was said earlier this morning about how negative data should be published and then the appropriate ways to include that in a publication. It's just like it might not necessarily be a large standalone publication, but it could be included with other data, or maybe even like a smaller publication. But I mean, the data is what the data is, but I don't think it should necessarily be reported until, again, it has been thoroughly investigated to make sure that this is indeed a non-reproducible result.
I think Keith showed how difficult it is when you think you have something that could be harmful to a patient population. It sure takes a lot of tenacity and actually a lot of time but definitely worth reaching out to some folks to get some other input.
Absolutely.
All right, I think that we'll go ahead and stop there. I want to thank the panelists. This was a good discussion. Just so you know, this will be recorded. It is being recorded. It'll be in under this particular workshop on the Rigor and Reproducibility website. We'll provide that to you in an email. Probably will come tomorrow along with a survey for today. Thank you again, Natasha, Kurt, Dan, and Robia. Appreciate you very much. We're going to move to the beginning of our afternoon sessions that will have breakout sessions. So while Melissa is getting ready, I'll introduce her here in a second. I do want to call your attention to the fact that you've received an email. You have an email that includes everybody that's in your group. We have 10 small groups today. These next two sessions will have small group discussions. When you are invited to join the breakout sessions, pay attention. Just make sure you know that you're in the group that you're supposed to be in. And you're going to designate a recorder and you're going to designate a reporter, because between Melissa and Stacy's sessions, each group will be called on to report out. We want to hear what kind of brilliant suggestions you came up with regarding these cases and questions. And then obviously not all of you will report out every time. So we want to have an email, a reply all email from the one that you received with broken down by session what your key takeaways were from your group. So yeah, with that, it was very, very fun this morning. We had a good panel discussion. I got to tell you, Melissa is also great. She does an incredible job making data management exciting. I don't know how that's possible, but anyway, with that, I'll turn it over. No pressure.
Thanks a lot. Thanks a lot, Suzanne. And thanks to the panel before. That was really excellent. I took some notes there as well. So hello, everyone. Thank you for joining and sharing 45 minutes with me. I do have that exciting topic of data management and resource sharing. So let's just dive right in. And any of you that are fortunate enough to be at home with some of your kiddos, I'll be doing a really brief little exercise, probably about 10 to 15 minutes into this, and they're welcome to participate in that as well if they'd like. So what I'm going to do is just talk a little bit about the principles and guidelines, move right into the data life cycle, talk at the end briefly about some things that you can think about to promote or excuse me, to ensure data quality and integrity on your studies. And then we'll do a really quick breakout session, as Suzanne was describing. One of the things I think it's important to remember is that you're not in this alone. There are typically what the community agrees on is 4 pillars of supporting robust science. And all of my links are provided in the slides, so you're welcome to read these articles a little bit more fruition if you'd like. This one is particularly interesting because it focuses on, as you see in this picture, how the institutions are falling asleep at the job and the researchers are looking to them for guidance and they're asleep. So while I'm not going to focus on that article, it's actually quite an interesting read. It is important to think that there are really stakeholders in this, and it's not just you as researchers, but it's also the funding agencies, the publishers, and the institutions. So the funding agencies, such as the NIH and the publishers, got together in 2014, and they had a workshop. And really the issue was to talk about this reproducibility, rigor, and research findings. And they had over 30 basic and preclinical science journals that were represented there, along with NIH. And that was really the journals that were the most prevalent in publishing NIH-funded investigators. The goals were to identify common opportunities in the scientific publication or publishing arena and to think about what they could do to further advance this and what might even be some guidelines and principles that the journals would agree to support or endorse for publication. The principles that came out of this and guidelines were the rigorous statistical analysis, which you've heard about this morning, transparency and reporting, that's a tongue twister, transparency and reporting, data and material sharing, considering established best practice guidelines for things such as images and biological reagents, materials, animal sources, endorsements that journals, associations, professional associations and societies could jump on board with, and then also adapted guidelines by these publishers. My job today is to really focus in on the data and material sharing. And from this point of view, we're talking about requiring the data sets to be available where it's ethically appropriate upon request. So as Dr. Ellis stated earlier this morning, that the real estate is quite expensive and precious when we're talking about publications. So it's not always, you're not always able to put in as much detail as you'd like. So if you can't, then it's important to say, where could this information be found? And sometimes this will be asked during the manuscript review. I've been through that myself. And then of course, upon publication for your peers. And then recommending that your data sets be deposited in public repositories where those are available, and encouraging the presentation of all the data values in a machine readable format in the paper or in supplementary information. Excuse me, there's also a requirement for the materials to be shared after publication and the encouraged to share software and a statement in the manuscript describing if the software is also available and how you might obtain it. So I'd like to put this link in. We don't have time for me to share this, although I wish we could. This is a really fun thing for you guys to look at or maybe even do during a lab meeting. It's kind of comical because it's in a comic, but it's really dives in and be quite serious. There's, let's see if I can do, there we go. So we have a clinician here that's represented by the cute little panda and a researcher that's represented here by the cute little bear. And they go through these scenarios where the clinician actually reads the research that was published and wants to use that with their patients and calls the researcher. And through a series of events, you find out very quickly, you go down this rabbit hole of where the researcher thought the data would be available. When he tries to find it, it's really quite difficult. And a lot of it is tied to a postdoc who's left. They can't find the postdoc, they can't find the code, and all those sorts of things that commonly happen in a or can happen in a research laboratory. And we laugh because we relate to them, but they really are potential problems, because the goal for all of us is really to see our hard work move science along and eventually end up improving patient health. So with these adaptive guidelines that I spoke to earlier, This is really the guidelines that the journals then bought onto. And I put a link here in case you'd like to see more of these. But just to focus in on the data transparency, it's really that disclosure. Are your data available? How can you share your data? So sometimes you can't share all of them. If you think about if you're working with clinical samples and you've got some patient information that's associated with that, and you've got HIPAA regulations, then what would you need to do to protect that? So those would be conditions for sharing that you would need to state up front. And can those results then be replicated using your data? Here's just an example. I always like to show examples of when things work properly. So the CRISPR technology has been in the news. I think if you've noticed, we have Nobel Prize winners around the development of that. And so this is just an example of where a large data set was put into a repository that were supporting the figures, both the figures and the tables, and that if anyone wanted the code for the plotting of those, you could contact the author and they would readily provide that. So for resource sharing, remember that if you're taking NIH or really any other type of government funds, you have a requirement or a condition on that funding that you have to be able to share that knowledge, because the whole idea is as a scientific community to advance that research. So remember that that's an obligation of taking those public funds. And resource sharing really goes beyond data. It includes anything that you're generating from those studies. So that could be samples, it could be critical reagents, it could be a model such as a model organism such as a transgenic mouse model. But what we really are going to focus on the rest of this presentation is on data, but some of those principles could be extrapolated when you're talking about sharing and conditions for sharing. They really could be extrapolated for what those conditions might be for whether you're sharing samples or reagents or even a mouse model. I found a really nice definition of data on the University of Pittsburgh website, and librarians are a huge resource when it comes to data, particularly large data sets and electronic data. So data means recorded information, regardless of the form or the media on which it may be recorded. The term often includes computer software, computer programs, databases, and documentation thereof, records of scientific or technical nature. It doesn't really include your financial award, those types of things, because generally in academia, there's a business or an office of sponsor programs that will maintain all of that. So they're not interested in the cost or the pricing, but they are really talking about your scientific data to include both your intangible data, such as your statistics or anything you do to data manipulate, your findings, your conclusions, your analysis, in other words, as well as your tangible data. And your tangible data is a really large net, depending on what type of research you do. So oftentimes we get folks that attend these sessions of ours, and they're in a variety of different scientific fields. So just to name a few, it could be notebooks, whether that be field notebooks or laboratory notebooks, printouts, electronic storage, photographs or images, slides, negatives, films, scans, audio radiographs, gel blots, etc. All of these are considered important data points in documentation. So where do we begin? Because that's a lot. I find it really helpful if we think about it in terms of a data life cycle. So if we think about the data from the generation of the data or the collection of the original source data all the way to the end. So if we start here with the collection, we think about where we are going to collect this data, how do we obtain this data, how are we going to be stewards of this data, where are we going to store it, how are we going to organize it, how do we protect it from being overwritten, then how we're going to analyze it, what methods do we use, what formulas do we use to analyze, where do we retain it? This even includes on a daily basis, where do you keep your lab notebooks on a daily basis? What would our conditions be for sharing and the subsequent reuse of that? And while I don't really talk to it in this particular presentation, we shouldn't be remiss in thinking about the disposition of data. And that's generally in academia things that we don't think about because that's set by state law, particularly in the state of Texas, and our records retention have policies around that. But if we think about cradle to grave, that's really sort of what we mean by life cycle. So sharing is part of the data life cycle. So let's first start about talking about data management plans. Again, from the University of Pittsburgh, a definition which I really like: research data management is a term that describes the organization, storage, preservation, and sharing of data collected and used in a research project. And it really focuses on the fact that data really are a scholarly product. They're fragile, they're easily lost. There's, as we've heard and discussed ad nauseum so far, and you know, there's growing research data requirements. Good management then helps prevent errors and increases the quality of your analysis. It really helps you troubleshoot when things go wrong. And as we all know, we learn from our errors, we learn when things go wrong, and we really want to be able to hone in on what actually went wrong, and we don't really want to have to spend a lot of time focusing in on something that was easily preventable, such as a transposition error, for example. Well-managed and accessible data allows others to validate and replicate our findings, and research data management facilitates the sharing of research data, and when shared, this data then can lead to valuable discoveries by others outside of the original research team. It's important that we think about what our data description, you know, what are the types of data we're going to collect? What are any standards that are around that, or format or content that we need to think about? Mechanisms for sharing, which include provisions for privacy protection, confidentiality, security. And as we move on in our research, we have a lot of different funding mechanisms. So we talk a lot about public funded and NIH funded, but there are pharmaceutical companies that will fund your research from time to time. It's generally advanced research. And so you may be working with a compound that you're testing, say, in an animal model or in your cell culture, and there are some intellectual property involved there. Or for those of you that are students and you want to think about working for a major pharmaceutical company or go into R&D there, that first capture of documentation is often associated with patents. So documentation then becomes extremely important. And then provisions for data reuse and redistribution. So let's move on to collection. As you're collecting your data, prospectively sit and think about what will your source data be, all of the source data, or we'll call that raw data. Sometimes you'll hear us say that. Excuse me, whether your data will be observational, will it be derived? What are those types of data? Will they be images? Will they be computational? What's your format? How much of it will you have? Are there any regulatory, such as HIPAA requirements you need to abide by? Any proprietary rules? And I'd like to ask, who's the owner of the data? At least when you're thinking about who is the one that's responsible for protecting and collecting and indexing and organizing the data. If I were to have a question about a data, who would I really go to? Who would I go to ask a question about that? Who's that owner? Who's always going to know where to access those original sources? And that source data comes in all kinds of shapes and sizes. It can be from the first time that you write on a plaque assay on a plastic well plate. It could be when you're writing on a cell culture with a wax pencil. It can be a printout here. It can be when you're writing on a tablet. It can be an image. It can be a field note. It's the first time that that data is written or it's captured. And this includes, I hope some of you are laughing, but this is very true. We grab the first thing, including at my desk, we grab the first thing that we can write down. So that could be a paper towel. It could be a sticky note or a Post-it note. Sometimes it's easy, as shown in this archaic picture, where we've got some tape here, where we've got gels in here. Sometimes it can be, just transferred and taped into the records. But what if we're transcribing the data? So in this particular instance, I show a hemoglobin value, and my raw data, my source data shows that it's 13.6, which for those of you who are familiar with that, that would be a normal value for, or could be a normal value for a particular animal. And if we transcribe it into a data set such as Excel for further analysis, and we miss the one and it's 3.6, we've now made a normal value into an abnormal value. And that's absolutely going to impact the data quality and integrity in our analysis. So I tell people, save your original source records. because you want to always be able to go back to those and catch something that could be very simple, such as a transcription error. Think about things prospectively. Where are you located for your organization and your storage? Where are you going to store these things, whether it's electronic or physical? If you have a lab notebook, where do you put it at the end of the day? Do you just close it and leave it on your lab bench that's next to a window? Or do you put it in a fireproof fire cabinet overnight or over the weekend, just as an extra source of protection? How are your data going to be transcribed? What's the accessibility? So do you have a electronic file that you save with your research team, or you share with your research team? Who has access to that? Is it just limited, or is it to a broader group who could inadvertently lose a file that's important? And how do you control for changes? So one of the things that we're using at UTMB is Microsoft Office 365. It's great because I can share my files with people. I can share my files with other researchers, with my team, particularly in the age of COVID. But when you do that, you notice that this autosave is often turned on, and on an Excel spreadsheet, it's going to automatically rewrite or overwrite what your information was. So if you happen to do, as Dr. Baggerly was saying earlier, you happen to accidentally start entering your data in the wrong column, or you're off by 1, then it's automatically going to be saving that every so often, and you're going to rewrite your data. So you have to really think about, do I open up a new file and save it with the date so that I can not inadvertently lose something that I had prior? Simple things like that can really make you pull your hair out if you don't think about them. Managing electronic data. If you have the ability to turn on an audit trail, a lot of those will have an on-off function. It does take more memory space, but it will maintain a lot of the data which might be important for you to reconstruct later, such as who collected that data point or who changed that data point if I were to have a question about that. Same things with security and encryption. Software compatibility is a big one. Backups are a good one. And really, I also want to think about program updates. Sometimes our institutions will push a program update to be automatic. That happened to me once when I was way back in my early career in a PCR lab, set my PCR up to go overnight, and the institution pushed an automatic an automatic update, restarted my computer, and I lost all of that data. Same way with the impact to significant digits. I've seen this on Excel, for example. And then data migration, if you're doing a research project that lasts over, say, a couple of years, and you're working off of GraphPad or Prism or something, and you get an update to that, what assurances do you have that your data that you collected on the old version is going to migrate over to the new version without any changes? And for those of you who use a lot of large data, there's a simple checksum application. You can do a quick Google search and find that. It's really nice. A lot of the folks that I know that work with large data sets will do that just to assure that their data files haven't changed. Quickly, here's just an example. You guys can go back to this if you want. They're just some examples of how you might implement a standard file naming system in your lab here. And on the right is just maybe if you're organizing by study, you think about those electronic files being named the same way and organized the same way, so it's easy to find things and easy to retrieve. Moving on then to data analysis, think about those methods that you're going to do to reduce your transcription errors. Defining inclusion and exclusion criteria. This is really important, and the previous panel talked about that. I see this all of the time where people, scientists will have a gut feeling that a data point is wrong, and so they'll tell somebody in their lab, well, just rerun it. That doesn't look right. And they don't necessarily take the time to go in and determine why that doesn't look right. They rerun it until they get the answer that they expect to get, and that's bias. So if you prospectively say, I'm going to do this in triplicate, and 2 out of the 3 have to be within a certain amount of statistical plus or minus whatever, then you set those parameters prospectively as opposed to retrospectively looking at something and trying to retrospectively fit your hypothesis. Prospectively, just develop a statistical plan. This is another big one. Love your statistician, talk to your statistician, talk to them to make sure when you're planning your experimental design, that you have the significant number of subjects to even be able to have your deductions matter. And then retain your methods to allow for your study reconstruction. So in the impact of time, or thinking about time, I also wanted to say you can go to this Office of Research Integrity, which Dr. Ellis talked about earlier, and they give you some really cool examples of image manipulations. So in this particular one, these are actually the same image, but this has been cropped, it's been lightened, so it looks like there's a tail here, and they've showed this is the control and this is the protein A, when in theory they're the same image. And this one, image 1 and image 4 are actually the same, and they've been flipped. Same way with the gel. So this happens a lot. And this is sort of just some general guidelines that the Office of Research Integrity say, which is document all of your changes and retain those unprocessed images. So again, that's your source image, is your source data. You can always go back and follow your journal guidelines for your permissible processing. So if we think about retention, it's just where are you keeping your files? What's your accessibility? Is there any security? What's your backup? How do you think about data migration? And defining your conditions for sharing, this could be a whole lot of different ways. You might have a small data set that you can say, well, I'll just email it to you. Or that might be how you'll provide the code, is through an email. So it may even be a combination of things. You might have an online repository that you can provide a link to. You might have a supplement to a publication. You might even put together a sharing agreement. This is really important for things that are confidential that might say need HIPAA requirements. So you'll have some very strict conditions for, yes, of course I'll share that data, but here are my conditions for sharing. You might have mixed media, exclusions, and maybe even instructions. So especially if this is an online repository, you might say, I'll provide the link and I'll email you with the instructions on how to get there and what my data set looks like. NIH even has, there's a link down there, a template for just sort of thinking about through what a data management or data sharing plan might look like, what your data are, who you could share it with, where the data will be, when they'll be put there, and how you'll have access and things like that. For reuse, you want to think about those conditions, any data that might be excluded from that, special instructions, how data might differentiate from the original, particularly if you're making copies, and how they might be retrievable. So I want to quickly do this data quality and integrity. I need to finish this up in about 3 or 4 minutes so we can get to my case study. But data quality, I like to think of in terms of Alcoa principles. And that's your data quality. Ask yourself, look at your data point and say, is it attributable? Do I know who did it? Do I know who to go ask if I have a question? Is it legible? Can I read it? Is it contemporaneous? Because the sooner that we write it down, the more accurate and complete our information is going to be. So if I ask you, what did you have for dinner last Thursday? Could you tell me? And the same goes with if you're in a laboratory. The quicker that you can write down the information, the more complete it's going to be. Original is important. So those are those original source documents or an exact copy of that original source document. So I can always go back to it. Sometimes we don't know why something fails in the lab until we read something or in the literature, or we have more scientific knowledge. And that's when we go back and say, oh, that's why it failed. That's why I can now move on. And so our memory needs to be written. And then our data integrity really is overall, all of our documents, all of our data, are they complete, consistent, enduring, and readily available? All right, so really quickly, I'm gonna ask you, pick up a pen or a pencil or something in a piece of paper. It can be anything. It can even be a Post-it note, because it's not an original lab data. And if you've got a kiddo, they can play along. So I'm going to put a picture up, and in 30 seconds, I want you to describe this. Tell me what this mouse looks like. Is it eating? What's it eating? Does it look healthy? Bright-eyed? What's the fur look like? Is it attentive? 5 seconds. Okay, stop. So now, if you think about it in terms of, look at what you just wrote down. Is it attributable? So did you initial it? And if you didn't, go ahead and take the time to do that. Is it legible? Can you read it? And so if your child was part of this, you might have a good laugh to say, can I read it, or can they read yours? Is it contemporaneous? Well, if I'm looking back on this, I would need to know what date it is, right? So did anybody date theirs? If you didn't, please date it now, quickly. So everybody should have a nice little description of the cute little mouse, and it should be initial and it should be dated. So take a look at how you wrote your date. And this is one of the things I find in research labs, that depending on what culture you come from, some people will write month, date, and year, and some people will write day, month, and year. And so if it's May 7th or July 5th, I have a problem. So you want to think about what are those consistencies. I just tell people use the initials, use May, M-A-Y, August, A-U-G, and then I really don't care about the order. But those are important for reconstruction of activities. Here's a really good reference. The WHO has a great reference out on quality practices and basic biomedical research, talks about making a full record of all your information to enable your study reconstruction. If we go back to that definition of data that the University of Pittsburgh Library said, remember, scientific data include both your intangible and your tangible data. So Dr. Ellis talked about this article that he was a co-author of. Remember that there were only 6 that were confirmed, and of those 6, these were the 4 commonalities of them. The experimental design included controls, verified their reagents, addressed investigator bias, and described the complete data set. Another thing I wanted to point out is communication is also very critical. I had an email come across my desk last year, and it was really quick. It was somebody who said, I'm going to miss the March 3rd Friday call-in. And if you look at the date, if you look at a calendar for March of 2019, the 3rd isn't on a Friday, it's on a Sunday. And what they really meant was it was a reoccurring meeting that occurred on the 3rd Friday of every month. So they really meant, I'm going to miss Friday the 15th. But the way they wrote it was, I'm going to miss the March 3rd Friday call-in. So think about what might be clear to you, but what might not be clear to somebody else. So my closing thoughts are, get organized. Remember, data stewardship starts with you and goes throughout the data lifecycle. Put together a really simple data management plan. You can put together a simple one that's probably going to be applicable to more than one of your research projects. Prospectively plan, as you've heard throughout the morning, and implement those Alcoa principles. Ask yourself any data point that you have. Is it attributable, legible, contemporaneous, original, and accurate? Okay, and with that, I guess I will stop sharing. Well, let me see, hang on in just a second. You guys are gonna go into different rooms. Make sure that you make note of the room. You're gonna have 10 minutes, and we're gonna put into the chat, this is gonna be the question. You're gonna identify options or the conditions for sharing data from a study of 500 human subjects being screened for sexually transmitted diseases. So just give me 3 things that you could do really quickly, write them down, and I'll ask maybe 3 or 4 of you guys to report out in 10 minutes, 3 things you could do to be able to share that. So I'm gonna stop sharing my screen, and I think we'll break out into groups. Is that true? Aha, so there you go. So make sure you know which room you're in, and I'll talk to you in 10 minutes. All right, thanks to all of you for your patience. This is the first time that we've done Zoom with like 100 people. And so the back end administration is really sometimes problematic. And thanks to Dawn for trying to work under pressure, because I kept saying, are we on yet? Are we on yet? So no pressure there. But I don't know whether anybody had the ability to actually be in a room for a while. Is there anybody that just wants to volunteer some thoughts? I see Chelsea raising her hand. Can you unmute? No, I see Target.
Sure. So in our group, we talked about a few different factors that we want to make sure would happen. So within that agreement, making sure that only the persons involved in the agreement would have access. So they're not giving the data out kind of at random. You might also pre-specify the variables of interest or your specific hypotheses. So that's pretty common amongst publicly available data sets. Make sure that it's de-identified information to the best of your ability, that there's no connections between who the people are, that it's for research purposes only, and that it's on an encrypted server so the data won't be easily hacked into. Those were some of the thoughts we had.
Those are really good. Yeah, look, see, you're getting an applause from Rovia. That's great. I think I have time for one other group. Anybody else want to unmute or wave their hand?
Looks like
Andrea?
Andrea.
Yeah, so we also talked about keeping the patient samples in the same folder or location so that when you were sharing with other people, they wouldn't get mixed up or anything like that. But basically, we said all the other same things as Chelsea did.
Wonderful. All right, so thank you so much, and thanks for being, brave enough to speak to everyone. I'm going to share my screen just to close it out.
While she's sharing her screen, I do just want to jump in and say, be sure you respond with, if you weren't called on, have your recorder send the email. You can send just one email because you'll have the next case study as well, but be sure and send the thoughts that your group discussed to us by email.
Great. Thanks, Suzanne. So I did want to say that I didn't make this one up. I actually pulled it from NIH. They have some really good examples. So again, if you're ever looking for something to do in a lab meeting that just sort of breaks the monotony, but also helps change the culture within your group, these are really kind of fun to do, and they take 5 or 10 minutes. So this is one of the things that, and there's no right or wrong here. Everything that everybody said was absolutely on target. So I'm glad you're thinking. This is just some responses that NIH posted, and they said, under the data sharing agreement that provides for a commitment to use the data only for research purposes and not to identify any individual participant, a commitment to securing the data. So if you're sharing, then the individual that's receiving it needs to sign that they are going to secure the data on appropriate computer technology, and that they also agree to destroy or return the data after the analysis. So again, you're the owner of the data, the original data set, and they would sign that they would agree to return it. So those are some things that you could do as conditions for sharing. So I wanted to say thanks very much. I appreciate your attention. And if any of you would like a really, really good resource for keeping laboratory notebooks, I try to share this as much as I can. And there's a link there. It's just a really nice, easy reference for best practices for keeping lab notebooks. So thank you very, very much. If you have any questions, just send me an email, and I appreciate you very much. Thank you.
Thanks, Melissa. As always, it's great. So yeah, it was kind of fun to see some faces, huh? I mean, you got a real clap from Chelsea, so.
Great, thank you.
All right, so now we're going to turn it over to Stacy, who is going to talk about publication and reporting.
All right, can we see the screen?
Yes.
Okay, and in present mode, right?
Not yet.
Uh-oh, it's showing on my, hold on, let me see if I can do it on the other screen. No, we're gonna do it on screen 3. Okay.
There you go.
All righty, so want to thank everybody for sticking around today. I greatly appreciate it. And so today what I'll be talking about is basically the publication and reporting aspect of our rigor and reproducibility, right? And moving from avoidance to transparency in research. And so this is just a hypothetical. I want you to think about it for a second. So I have a question to you. So why do we really loathe publication and reporting? So I want you to think about some examples in which you have personally exhibited avoidance behaviors. So things like procrastination with respect to publication and or reporting in any of your current or past research projects. So think about what might have been going on that just led to that avoidance behavior, all right? And we can definitely share more about that later on in the presentation. And the big question is why? Why do we have these avoidance behaviors? And so typically what this is that it's avoidance motivated behavior basically, right? And so our behavior is this form when we distract ourselves from doing a task that is associated with some type of unpleasant emotion, and that emotion is typically fear. And so it is also known as approach avoidance. And so there are three main types of these behaviors. There's complete avoidance. And so examples of this would be no showing at a presentation, failure to submit documents, or to quit a responsibility. Another form of behavior is escape, right? And so this is where you leave early from a commitment, you end a talk abruptly, or you hide in your office. So yes, I've heard of stories of people hiding in their office so that they didn't have to perform some task. And then lastly, there is partial avoidance. And with partial avoidance, these are situations such as daydreaming or avoiding eye contact. And the main feelings that drive this are things such as worry, anxiety, and panic. And when we perform avoidance motivated coping strategies, these behaviors, they don't actually reduce our anxiety. They rather tend to fuel it or cause difficult situations to snowball. And so they can lead to worse situations later on. So what does this look like? So the number one way is procrastination. We do something else to give us momentary relief. So there's a lot of examples of this in PhD comics. And so here we have basically the chart in which we map procrastination onto what you should be doing. Another example of this is there's the field guide to procrastinators, right? And so you basically get to choose what type of procrastinator you are. Maybe you're a cleaner, maybe you're a snacker. In both of those, you know, some people will basically binge watch Netflix. Another funny example of this is the 4 horsemen of modern procrastination. So napping, snacks, social media, and minor chores. And so here you can actually see them standing behind our scientist and beckoning him or her to basically move away from being productive, right? So it's just a funny cartoon. And so in terms of avoidance motivated coping, when we're trying to do something else to bring us momentary relief, for some individuals, it actually goes much, much further, much, much darker. And so that in order to get out of their responsibilities, they might do something such as data fabrication, such that the data were never collected, and then they just make it up, or data falsification where the data may have been collected but are illegitimately altered. These are extreme forms of avoidance motivated coping, and I hope that nobody here ever does that, because those are very serious maladaptations, right? These go against our ORI principles that both Keith and Lee spoke about this morning. So why do we exhibit these avoidance behaviors in publication and reporting? Well, one of the biggest things is fear, right? We have a fear of failure. And sometimes there's a lot of pressure. And this is something that has been very consistent throughout the day, talking about pressure to publish, right? Have those high impact publications. There could be not just that, there could be lab pressure or peer pressure. There was a situation that was discussed earlier this morning in which 2 students or postdocs would be pitted against each other. And so whoever came up with positive results first would win. And sometimes some students and postdocs or even investigators might have employment or visa pressure, right? So there are different forms of pressure that individuals can experience. Other situations that might lead to these avoidance motivated coping mechanisms. Some individuals just have difficulty with time management. You know, when you move into more administrative roles, and I'm going to show like a funny pie chart about this. Basically, when you move more into administrative roles and more writing heavy roles, those pieces can be very costly in terms of time. And especially earlier on in your career, you can have a lot of difficulty managing that and just realizing how much time some of those things are going to take, right? And so you might see them as time sucks, but they are necessary things that have to be done. And sometimes deadlines that you have can create time conflicts with other commitments, right? And so it could be teaching commitments, it could be family commitments, service commitments, travel, maybe not this year, but in years past and in the future potentially. So issues with time management can definitely be a problem. Other forms of issues that might pop up, generally feeling a lack of progress, right? And so often we feel like we haven't been productive enough, right? And so that's the funny cartoon at the bottom of the screen that says you should be writing basically every single hour, right? This feeling sometimes can be paralyzing. And there could be a lot of reasons why you're just not making progress in your writing, right? Maybe you're having difficulty in acquiring needed information, or just even progress in your own experiments if you are having difficulty acquiring materials. And sometimes progress can really feel like a moving target, right? So we know that these avoidance behaviors are negative, and they can definitely impact publication reporting. You can just basically miss your deadlines. You can have delayed publications. You can be submitting late reports to regulatory and funding entities, which is a problem. It's a huge problem. And all of these things can snowball into problems with career advancement, promotion, and tenure. And so if we procrastinate, it can make easy things really hard and hard things really, really hard, right? So what types of things are we talking about in terms of reporting? So basically, publications are the most commonly sought after products, but there are other products such as conference papers, podium presentations, abstracts. It could be, technology briefs. It could be patents, patent applications. For those of you in biotech, it could be licensing agreements. It could be other forms such as databases or physical collections, software, instrumentation. It could be any number of things, right? So it's not just like one publication in Cell, Nature, Science. So who typically does the work? And so typically your PI or your co-Is are going to be your senior authors, but the people that really do the heavy lifting are typically your postdocs, your research assistants, your students, undergraduate students in your laboratory techs, right? And so there can be basically bottlenecks at each one of these levels. The PI is usually supervising, but they are typically supervising multiple projects at once. And so the junior scientists, right, the trainees are typically doing a lot of work. And so when we have avoidance, we can have avoidance at all of these levels, right? And so if you have avoidance early on, sometimes they can just stack on top of each other. And then we just get these massive delays in getting these projects done and out there. So different types of research reporting. And so if you just think, oh, it's just publications, just my publications late. Well, that's not necessarily it, right? There's, you might have to report back to your department, your center, your university, what exactly you're doing. IRB, for example, you have to report to them, or IACUC. If you have grants, you will have to submit annual reports to your funding sponsors, right? So NIH, NSF, could be local foundations, could be national foundations. They're all going to want to know basically how you're doing on your projects and how you're spending your money. And so basically when you're more junior and basically as a student, as a postdoc, some of these more administrative pieces you might not be so involved in, but as you transition into more PI roles, you're going to have a lot more competition for your time because you're going to have all these different types of reporting and just administrative roles that you have and administrative hats that you're going to have to wear. And so this is key to that time management issue. Just things that you should be aware of, research performance progress reports. And so this is a format that's basically, it's the NIH format, and a lot of other entities have adopted something similar, whether they are at state level or just private foundations. It's typically required on an annual basis. They can be required even more frequent, such as every quarter or every 6 months. And typically only the PI or a noted delegate that's a representative of the institution can actually initiate this. And so this can take a lot of time for your PI to do. And there's a lot of pieces that can go into it, right? And so for some institutions, you have a lot of manpower and resources to deal with these things. You might actually have an institutional representative that handles all this for you. Other institutions, not so much. In some cases, the PI will be doing this all on their own, particularly if these are more for foundations. So for example, at U of H, you know, some of the smaller foundation grants, the PI would be doing all of this. Whereas for an NIH grant, we have institutional delegates for this. And so what components might they be asking? So it's not just, oh, simply I did X, you know, give me another $200,000. That's not the way it works. So basically you have to list your accomplishments, you know, what were the major goals and objectives of the project, and, you know, what progress did you make towards these goals? Were there any opportunities for professional training for your mentees or for yourself? Did you disseminate any of your results, whether they were at conferences or at just, you know, community presentations? And what do you plan on doing for the next year, right, to support the goals of the project? They'll probably also ask if you had any publications or products, you know, things like patents, databases. Do you have any preliminary data to show? Do you have any patient or animal recruitment and safety reports? You might have to disclose how many subjects that you were able to recruit, you know, what race, ethnicity categories they were in. If there were any adverse events, you might have to report those and have any documentation on that. If your projects included collaborative efforts, you will have to share information from the other institution. So basically, if you have PIs or co-Is at other institutions, they're going to have to create their own reports and then send to you or your institutional delegate that then gets sent on to the funding body. They can also ask, you know, what impact are you having on the field? Another big thing that you might have to report are project changes, challenges, and problems. If you had to pivot or, you know, change an approach, why did you do that? How were you able to solve it without doing like a budget overrun? If there's any anticipated problems that you're seeing. So COVID is a huge one right now. A lot of us who do human subjects research, we can't interact with human subjects because we need to be close contact. So that's a major situation that we're dealing in terms of project challenges and problems in the times of COVID. And that is definitely impacting our expenditures. So that's something that we would have to report. And if there's any significant changes in the care of human subjects or vertebrate animals, biohazards, so that's something that's definitely COVID applicable at this point. You'll definitely have to have some type of financial or budgetary report. And if there's any modifications, you have to report that as well as future plans. So there's a lot of pieces to these reporting aspects that you might not think about earlier on in your career, but you would be responsible for later on. And so when we do these reporting pieces, but also in terms of our manuscripts and conference presentation, there are some core standards that you should be including in these reports. So for example, if there's randomization and how that randomization has occurred. So this is something that you should be looking for in terms of like when you're looking over manuscripts, when you're looking over grants, conference proceedings. These are some of the things that you should be looking for, right? Randomization, if there's any type of blocking factor going on, and block randomization, for example. You should be looking to see if blinding was done, and if blinding was done, was it a single, double, or triple blind? You know, allocation concealment in terms of those who do animal studies, you know, was that considered or even discussed at all? The blinded conduct, like I said, as well as a blended assessment of outcomes. So that would be like your triple blind, right? And so where the subject is blinded to the treatment, the administrator is blinded to their treatment, and the person that's analyzing has been blinded also. So just different examples of blinding. If it was used, that should be described and how that was ensured. What else? Hopefully sample size estimations have been provided, particularly in grants. An appropriate sample size should be computed and utilized. So make friends with your statistician. They can definitely help you a lot on this. And your variability measures, right, should be included. So here's just an example I pulled from one of my projects where I basically showed the different measures under each aim. And you know what I use for the two different groups, my alpha levels, my beta levels, as well as how many subjects we would need per group to show statistical significance, right, at 80% power. You'll see that I have under aim 2, that one had the largest edit, and so basically you need to power to your weakest aim, right? Because if you're underpowered for your other aims, that's going to be a huge problem. So that's something that you should also consider. In terms of reporting, you also need to be very careful as to you just correctly and accurately reporting your data handling techniques, right? And so what rules did you set up for stopping your data collection? What were your inclusion and exclusion criterion? How are your outliers handled, or any data exclusion? You need to identify your primary endpoints for data collection, as well as how missing data were treated. If you did any type of pseudo-replicate analysis, you need to be dealing with that, as well as if you did replications and repetitions under a range of conditions, or if it was the exact same every single time. So these are things that not only you need to report, but you should be looking at in literature that you're reading, and this is outlined by the Landis paper in Nature. Okay, so there's all these things that you should be doing, right? And there's a lot of challenges for you to do this, right? You could have problems getting your lab up and running, or there's something broken, or there's just a lack of time, or in this case in COVID, maybe you can't even get to the laboratory or bring subjects in. Maybe there are just deadlines that are just conflicting, and you might have issues with institutional support. When you do become a PI, you need to know what the institutional support is to support you, so that you're not trying to lift everything on your own. If there's help, please use it. Moving on, I want to talk about, if this is so tough, why do we even bother with it, right? There can be significant negative consequences if we don't take the time to do publication and reporting correctly, right? They can be personal consequences, such as withdrawal of funding, and inability to support your current and future projects, but also longer term, right? It could be your own career, your own promotion, your own tenure, as well as those of your lab, basically, right? It's going to impact your students, your postdoc, your research assistants, your lab techs. Other things that you need to think about. So it's not just personal consequences, but also consequences to the field, right? And so when we have this lack of reporting, we can actually accidentally introduce bias into the evidence base, right? And the biggest bias that we see is reporting bias, which is the selective revealing or the suppression of information or outcomes of a study, where you report some results, but not all of them. Or spin has, quote unquote, spin has been applied to unexpected or undesired results, right? Where these undesired results might be attributed to sampling or measurement errors. You need to be very cautious when you see these things pop up in manuscripts. Other types of biases that might pop up due to a lack of publication reporting, publication bias. And so essentially this is non-publication of results. Typically this is due to some type of failure of an intervention. So this is your lack of positive or significant result reporting. There can be time lag biases. So basically rapid publication of exciting results that aren't full, whereas negative findings can be delayed or just even never published. Or a PI might try to be gaming the system and try to boost their productivity under a specific grant. So they're basically doing almost like a salami science, publishing different studies at different points in time. Other issues, citation bias can definitely creep in. We tend to cite positive findings more frequently, and that can lead to a perception that an intervention is more effective than it truly is due to a differential number in your citations. And so it's just an overrepresentation of positive findings. There are other issues such as language biases. We tend to publish positive findings in a specific language, specifically English, whereas non-significant results are sometimes published in non-English language journals. And that definitely impacts things like meta reviews and systematic reviews. So that's something to be aware of. Other biases include outcome reporting bias. And so this is your expectation bias, where you selectively report your results and you modify your hypotheses to fit your findings, right? And so we know you shouldn't do that. And here you also have, you are less likely to report adverse outcomes. Other issues, location bias. So this refers to journal of publication and impact factors, right? And so this is your CNS, so your cell nature science bias, basically. So where studies with positive and significant results tend to be. And so, you know, the funny cartoon down there is basically like the modern scientist, you know, can I get my paper into Nature? That's what this type of bias is. That's what location bias is. The last major type of bias I want to introduce is the funding bias. And in some cases, a scientific study, if it does not support what the sponsor wants, they can try to suppress the results. This is something that has happened, you know, in the past. Hopefully you do not experience this ever. Predetermined conclusions can bias researchers into their own expectation bias. Some sponsors do require their PIs to waive the right to publish findings that don't support the interests. And so that just ties directly back into publication bias as well as outcome reporting bias. So you have to be very careful if your sponsor wants you to sign off on something like this. This isn't common practice these days, but once again, just be aware of this. But also some of like the funding mechanisms themselves could actually influence study design negatively and just be flawed from the start. And so those of us who are PIs, we have to be careful if we take money from different institutions so that it does not impact our study design, right? This is part of our COI reporting that we undergo. And so how can you reduce bias in publication and reporting? You need to build a case in which negative results provide a positive impact on the evidence base, right? It's all in how you frame the story. Don't try to suppress your results, particularly negative results or non-significant results. Don't try to rush or delay publication, and also don't dual submit. Dual submit is highly unethical. The next one, the publishing the same results in more than one manuscript unless you have a really good reason to do so, right? So that goes under the dual submit situation. And carefully consider the manuscript or the journal that you are submitting your manuscript to, right? Is this the correct audience for this? And try to balance the story. There are a bunch of guidelines that the NIH has actually produced, and there's a link within our website here, or there's the link within the slides, which will be available to you after the presentation. Also, don't withhold critical information and be organized. Also set realistic goals of your team and have authorship and team expectations set out early on, right? So who's going to be the lead author, who's going to do what in the different aspects of the projects. And so this can help with accountability and just making sure that your projects move in a timely manner. For those of you who are team leads, communication is extremely important, right? If there's something going on where you sense that there's stress or just something's going on with one of your team members, just check in with them, right? This is one of those soft skills that is really important in science. Check in with them and see what's going on. Is there something going on in their personal life that is really impacting their productivity? And is there something that can be done? Are there resources institutionally that can help that individual overcome whatever stressful situation they're in? Don't try to wait it out. So like I said, when we're doing our publication and reporting, we need to make sure that we are reporting basic core standards such as randomization, blinding, sample size estimations, and data handling. And so look for these things, right? So not only are you expected to produce them, but you should be looking and expecting this from other individuals. Also, as Dr. Eitzen said, please use the Alcoa principles for managing your data quality. So attributable, legible, contemporaneous, original, and accurate. And just make sure that you are maintaining your data integrity such that it is complete, consistent, enduring, and readily available. In terms of you as a PI, you need to know the strengths and the weaknesses of your team, right? And be able to familiarize yourself with the expectations of your editors, your review boards, your funders, and your institutions. That will help you actually manage a lot of the expectations that are of you as a PI. Know your deadlines. I live and die by my Outlook calendar. For those of you who know me, it's color-coded. I have all kinds of things on there. And also know the things that you are expected to produce, right? So different funders have different things that you would have to produce. Different regulatory agencies are going to have different things that you have to produce. Know what you need to do so that at the last minute, you're not rushing and submitting something that's sloppy or inaccurate, right? That just, it's not something you should be doing. But also know your resources. Know who can help you at your institution, whether it's members of your team, whether it's somebody within the research office that can put different pieces together for you, or grants and contracts that can help walk you through the process, especially earlier on in your career. So know what the resources are available to you, and that will save you a lot of time also. So I've presented some things for each of you to think about, but I have a challenge for you, the 2020 challenge. Every day a new year can start. So New Year's resolution. I want you in your breakout groups to think about what you and your lab can do in particular to improve your relationship with publication, reporting, and bias. And so I want in your group discussions for you to come up with 2 to 3 different resolutions and implementation strategies to share with the group, right? So when we go into our breakout groups, somebody needs to record this and somebody's going to have to be reporting out. So just some different ideas or different techniques that have worked for you, or that you've seen other people implement successfully. So that is in terms of my slideshow. Dawn, are we ready for breakout rooms?
We are ready. If I miss somebody, I will throw you in as quick as I can.
We'll still, even though we're a little bit early, we'll only do 10 minutes, and then we'll come back and report out and do the post-assessment survey.
Okay, here we go.
Okay, Stacy, I think everybody should be back. If you want to call on some folks to report out.
We're doing evens, right?
Yeah,
or just
well,
whatever.
it doesn't matter.
All right. What about group 8?
8 actually reported in the last one.
Oh, they reported? Okay. Well,
We had totally different people, Suzanne.
Yeah, I was in group 7 before, and now I was in group 8 this time.
Okay, yeah, call on 8 then.
Go, Emily.
So we were just talking about number 1, how important it is to do report negative data, and that's something that's not as done. So that was a big resolution. And another one is allowing the PI to be aware of data in all of the stages of its development, and not just like the pretty final results of what the data looks like. But I think we also are saying that previously it was talked about people giving PIs weekly updates on the data, and none of us had that personal experience in our lab. And then also talking about what to do when someone is having misconduct or doing poor data management, and how to actually mitigate that if the consequences for that person could be severe. So I was telling one example where I saw someone immediately fired as soon as they were found out that they had been, not intentionally, but a little loose with the data. So those are some things you talked about, identifying those.
That's great. Group 10. Anyone from group 10 want to report out?
I can go ahead and report out. We had so we had kind of discussed, let me put my video on. So we had discussed as incorporating into a live meeting setting of either like monthly or some type of occurrence, people actually reporting the data that doesn't quite make sense, or data instead of neglecting it and trying to push it off to the side, but kind of bring it to the forefront, as well as we, you know, if, for example, a scientist in the lab is reviewing and notices something within a publication they're reviewing as being misleading or not, maybe not being shown correctly, showing these as examples of, you know, maybe what to do, what not to do when we come into these problems we might see going through publications.
Nice. Excellent. Group 2.
Hello, I can actually report. I think we pretty much agree with the two previous groups that we want to actually encourage people to present negative data. And we also suggested that we can make a checklist of the things make a checklist, make sure that we stay organized and make sure that we know where our data is, and let's see. Yeah, so make sure where our data is and our agreements in data sharing and all that. But that's pretty much what we discussed. So
good. Yeah, I know there's been discussion of Slate channels also. That was one that came out in one of the earlier times that we had this. So use of different ways to organize and communicate among teams. I'm going to have group 4 report out before we wrap up here. Anyone from group 4?
I'm so sorry. I was speaking. I just was muted. So that's the end of my report. One thing we talked about was streamlining parameters. Most computational work has parameters, and it is our job to write these down as we're doing the data processing, but also justify why we chose these parameters and explore other ones and report that exploration when publishing. Another thing we talked about is like SMART goals. So basically we want to set short-term concrete goals that are achievable. Discuss these with other people, PIs, coworkers, just to sort of improve accountability. And number 3, basically regularly talk to your collaborators, workers, coworkers, PI, and ask the question, are you seeing what I am seeing? Because that's very important, and that's one of the true ways of dealing with sort of bias, getting other people involved.
Great, really great suggestions today. Thank you so much for each of you. It's really been great. I'm going to turn it over to Suzanne. We're right on time.
Oh, great. Thank you again to all of you, and a little bit bummed that the first breakout sessions didn't work super great. I think we did better the second time, but I do appreciate your patience and look forward to reading your emails, hearing from those of you that didn't get called on the second time, hear what your groups were talking about. In the meantime, Shannon, I'm going to go ahead and ask you to add the post-assessment link. As soon as you take the post-assessment, you're free to go. Please pay attention to your emails. You'll get a survey in the next day or so, and also a link to the website where you can find the resources and recording from this meeting. So again, thank you all very much. And yeah, we look forward to hearing your feedback and your thoughts.